Okay, we are live. Hello, everybody. Welcome to SOI Divestream 530. This is our second dive of our Octopus Odyssey Expedition, and we are off the Pacific coast of Costa Rica. We are just about to hit bottom. We're about 10 meters off. Bottom is about 3,170 meters. And I will introduce you to Sergio Cabanero of the University of Costa Rica, who will be narrating this dive. Take it away.
Hello, everyone. So this is Sergio Cambronero from Costa Rica here, and I'm pleased to join you this morning in Costa Rica, evening for some of you, depending on where you're connecting. And as Juliana said, we're about to get to the bottom. We will be revisiting some same area we did on our previous dive. And initially, we will be testing some of our heat flow probes, and we are aiming to do some transects over the abyssal plain. So stay tuned as we hit the bottom. Buenos días para los que nos sintonizan. Un gran saludo. Mi nombre es Sergio Cambronero y el día de hoy vamos a estar revisitando el sitio que estuvimos muestreando el día de ayer en nuestro buceo. Estamos en el Dorado Outcrop, mar adentro de la costa de Guanacaste, aproximadamente a 3,160 metros de profundidad. Como ven, nos están recibiendo esta medusa. Así que bienvenidos a la transmisión del día de hoy. Y nos gustaría ver sus consultas, preguntas, saludos. Las estaremos leyendo por acá. Juliana, is it possible we can have the same display as yesterday with the...
Yep, I'll bring up the chats
right
now.
Thanks. Is there any people? Anyone connected?
7.
Good. It's almost 6 in the morning in Costa Rica time, and we're about to hit the seafloor at Dorado Outcrop.
Bring it on up and just hold it there for a second.
As our ROV team finishes some details, we will go over some of the main objectives of today's dive. We will be first heading over the abyssal plain in this area. Then we will aim to go around nodule fields, and before we get to the outcrop, which is where we will find the octopus dense area that we were visiting yesterday. So for the ones who don't know, the main objective of this expedition is to understand better the heat flow, the outflow, hydrothermal venting associated with brooding octopus at Dorado Outcrop. That's what we're seeing. It's a sponge. What's that? What's that? Anemone? What's that? Owl. So these abyssal plains are mainly characterized by a thick layer of sediment. So all this sediment has been building up for years and years of particles coming up from the surface. And as you see, there is a lot of life down here feeding and thriving on this sediment. So we have here an anemone. Welcome everyone who's tuning in. We're just hitting bottom here at Dorado Outcrop, offshore Costa Rica. 6 in the morning already. And we are over the abyssal plain. Firstly, we will be testing some of our heat flow probes by our colleagues at U Calgary. Yes. You feeling okay for the testing?
Yeah, it's your power to that probe.
Yeah.
Okay.
Yeah, that's the first thing. So, what's that? Oh, that's the same sponge.
Heat flow probes on,
Huh?
on down.
Okay. Do we want to put it into the ground or just?
Yeah, just we can go over there. I think that area looks nice, and just try to aim for the same thing as yesterday, to see if we can have the better data on that last sensor. Huh. Did you see that, Diva? That circular mark?
I did not.
If you look in the sit camera, right there.
Amazing.
The anemone that buries?
It's
Maybe.
The one that walks. Oh, it's beautiful. Anemone ahead of us. Maybe that's what made it.
Mm-hmm. Yeah, maybe. So, as we go over this abyssal plain, usually what we look for, of course, is not only all the fauna you can find, but also the signs of any activity. So usually animals bury around these areas and they leave marks. So that will also tell us whether there is activity going on of different taxonomic groups.
Alright, so Ben, what we're going to be doing is we're gonna take the little poker and we're gonna push it in, set it in front of us, kind of hold it down there.
On the side.
There you go. No, you're gonna grab that one. Dig us in there, hold it in place, that way I'm gonna come off of that. That's what we're pitching. So I need to come over and back.
You ready to do that now?
Okay.
This expedition, Octopus Odyssey, is being done, composed by an international multidisciplinary team from colleagues from Costa Rica, United States, Trinidad and Tobago, Brazil, Portugal, and Canada. So what you're seeing on screen right now, it's what we call a heat flow probe, and the marks on that probe identify or denote the position of heat sensors, temperature sensors. So we will be introducing this into the sediment to assess the heat flux.
That one's for testing. One on the side, that's marks on it.
Okay.
We took the marks off of the one on the side.
Okay.
We were told that it might be interfering with
Okay.
readings, so.
Yeah, I'll be able to tell by the temperatures.
Good morning, good evening, the ones who are tuning in. We're over the abyssal plain southwest of Dorado Outcrop, which is offshore Costa Rica. Those of you who might wonder, we are pretty interested into knowing more about the temperature distribution and the horizontal and the vertical in this area, because we have collected evidence of hydrothermal activity. So this is why we're focused on particular temperature measurements.
There goes that.
Yesterday we had a great dive. We saw, I would say, over 1,000 octopuses, all kinds of different behaviors. So I don't have any doubt that today's dive will be also pretty spectacular. So stay tuned as we travel from the abyssal plain to the nodule fields and then to the outcrops.
Just want to make sure I'm clear. Probably have more stability if I get closer to us, right?
We'll just kind of like angle towards us. We're gonna put this like this. And then keep spinning. Hang on, yeah, hang on. Yeah, draw it to us.
Yeah, I'm gonna be back.
So what we're doing right now, and what you're seeing on the screen, is kind of poking the sediment to see if it's apt for our temperature measurement. And not only we're interested on the megafauna communities and meiofauna, but also on the microbial community composition around this area. So we have folks on board that will be taking samples from this sediment and different layers to assess and evaluate the not only the composition of these microbial communities, but also the potential ecosystem services that they provide to humankind. And this is very important for us as scientists as we try to translate our science into management and informed decision making. Are those the same polychaetes? The one, that thing poking there,
Oh, same as what?
as the ones we saw yesterday? Yeah.
Two
Big.
polychaetes, we wouldn't be able to know what's in now.
Okay.
But it looks similar.
Okay.
So I lowered that about that far.
Okay.
I'm gonna go ahead and put it in now just to see if it gets all the.
Yeah, so I'm recording right now.
Okay, so I'll go ahead and do it?
See, yeah.
All right. That's fully in or fully down. We are pitched that way. We are on the metal, so.
Juliana, can you, is it possible to pull the EK80 image on the screen?
Yeah, sure.
Yeah, thanks. Hello, everyone, just tuning in. Welcome to our second dive of the Octopus Odyssey Expedition. We are live from Dorado Outcrop offshore of the Pacific coast of Costa Rica. And it's about to be 6 in the morning, actually 6 in the morning right now. And the sun is rising, and this means that the diel vertical migration of zooplankton, which is the largest migration on Earth, just it's on its most intense point. As you can see on your screen, we have an image of our sonar readings, our EK80, and you can see a layer coming down from left to right that actually tells us about the movement of this huge biomass, planktonic biomass that comes up at night. Particularly yesterday was more shallow than in other times because of the full moon, we believe. And as you can see right now, they are going down, and this is usually associated with the sunrise. So this happens every day, and the readings we're getting will tell us and guide us to get calculations on the total biomass that this represents, and that at the end can translate into carbon, into what we call the carbon pump, the biological carbon pump. So we're not only interested on the seafloor data, but also what's happening up in the water column. So we have a question on our Facebook chat if specimens explode when they're brought to surface because of the pressure. So that sometimes it can happen, but usually it happens when that change of pressure is abrupt and fast. So usually in this type of dives, we don't experience that, or we don't see that in our biological samples, because the ROV has a very slow and constant ascent rate. So this is usually useful also to avoid any pressure differential that can affect the sample. So the answer is, for this type of collections with this ROV, no. And it will depend also on the biological, on the sample itself. We are at 3,168 meters right now. And what you're seeing on screen is our initial testing of our second dive of the expedition. We will be testing some of our heat flow probes.
Same thing as yesterday, but 5:30, right?
That's
This
right.
was your one.
So, yeah, dive might be.
And there's that, I think it's hard to like say
5:30.
like interesting stuff while we do this, because it's like pretty, it's different than when we do the kind of seeing a bunch of stuff.
And Sebastian's hand is actually a proxy for our heat and flow probe. We want to make sure that we don't hit any hard substrate when we're putting it in. So Sebastian's arm will take a stick of its own and push it in the sediment. So how far we can go down
Yeah.
before we deploy the heat probe on a hydraulic ram, which is shown in the
That's cool,
picture
yeah.
I hear you struggling
on
with
your
this
screen.
Okay, thanks. So welcome everyone who's just tuning in. We're just been down in the seafloor for almost 25 minutes, so the dive is just actually beginning. Buenos días a los que nos sintonizan desde diferentes partes del mundo. Estamos en el afloramiento rocoso El Dorado, en Costa Rica. Bienvenidos. Nos gustaría ver sus comentarios, sus preguntas. Por acá los estaremos leyendo.
Okay, we're good to pull it out.
That's good.
Yeah.
Good people. Nice.
So.
So we just confirmed that this site is apt for our heat flow measurements,
Are you,
which is something that we tried yesterday, and there was some modifications to improve our data collection. And fortunately, this dive will be getting better data, which is what we always try to do. In each dive, we try to improve our data collection methods. And again, all this data usually will tell us more about the specific dynamics of this site, not only at the surface but down deep in the sediment layer and in the ocean crust.
What would you do next?
Hmm?
What would you like to do next?
We're good. Rob?
yeah, we're good.
Good. Okay. So we were aiming to do some transects.
Okay.
And yes, so I will just give you a heading. That's our heading, right? 312. Yeah. So
How is our goal this direction fly here, or is it just there?
we just want to do random. We don't want to get too far from the from the track, for the goal tracks. But yeah, we will just give you random headings and
Okay.
do 100 meter length transect. So the idea is to do it at 0.03 knots, 1 meter per minute, and 1 meter off the bottom, if that's possible. And
Okay.
the idea is if we can have the camera fixed, and even if we see cool things, I was going to ask you if it's possible that I can give you a signal to, if we see something, if you can drop like a waypoint,
All right.
or if
Okay.
in
So-
case we might return later.
If we keep this one right on the seabed, and then you have freedom to move this one wherever you'd like, for something you want to look at, or we keep them both fixed. It's up to you.
Yeah, let me just double check, because I think is it possible that we can take the basket?
Yeah,
and-
yeah. That'd be great.
Yeah, okay.
It's just so we can see what we're doing.
Yeah. So Diva, how are you feeling about the camera um, the camera configuration before doing the transit? So this will, I know you mentioned yesterday that
Complicated to push towards that?
no, we will do that later. There was a little tweak in that.
Okay.
So we will be doing the push first on the nodule fields, or closer to that.
So once we lift off the seafloor, then we'll just adjust it. But we basically want the high resolution camera tilted as down as possible.
All right. That was a question if we wanted to fly, which yesterday.
Yeah, but yesterday, and it wasn't.
Yeah.
So that's why we stay with it. So basically tilt it down and then fix in that position. If we could sort of note it for future dives. And we're driving forward and not basically.
So welcome everyone. For those of you joining, we're down at 3,168 meters at the proximity of Dorado Outcrop offshore Costa Rica. We just finalized our temperature measurements, and soon we will be starting our video transects over the seafloor. So stay tuned, because we might see some pretty interesting stuff. as we see in our uh, social media chats, we're also really looking forward to seeing more octopus hatchlings today. Um, Yesterday, in our first dive of this expedition, we got pretty great data and footage. So we're looking forward to going back around this area and um, get more observations on the behavior and the physiology of this particular octopus species. So huge shout out to the Schmidt Ocean Institute team, because for us Costa Ricans, this is really valuable opportunities to explore the seafloor with this very amazing tools that otherwise we do not have available in our country. So special thanks to them for making this possible. And as a marine biologist and working in physical oceanography, also this is pretty amazing, as we have visited previously these sites, because usually in the water column you will find evidence of a oceanographic feature that's called the Costa Rica thermal dome. So this has been studied for many years because its importance on circulation and hydrography of the region, and also because it's a pretty important feeding ground for many species, such as blue whales that come down from northern latitudes to feed here. So these are very productive areas up in the water column. So we have collected previously observations at this point, but just in the first 1,000 meters of the water column. So for me personally, this is a pretty awesome experience to see what's down and seeing with this really high resolution on all the animals living here. So yeah, great opportunity. Ya vamos a empezar el concepto, sí. Acabamos de hacer las muestras de Heat Pro. Sí, la idea es empezar y hacer como los transectos random. Entonces, más bien, mejor si usted quiere decirles un heading, la idea es decirles como no tener transectos, ¿verdad? Como usualmente, como uno pensaría, paralelos, sino hay una línea al azar. Entonces ahí si usted quiere decir un heading, le decimos a ellos y vamos hacia esa dirección. Pues 45 grados para allá. Sí, digamos, la idea es ahí, ¿verdad? Esa linecita verde es como la que deberíamos seguir. Entonces la idea es como no desviarnos mucho, pero sí, yo creo que podemos hacer 45 grados, luego 100 y así. Sí, ajá, ok. Sí, como ir haciendo así, como. Ok. Sí. Una esponja ahí abajo.
lo sostiene son fibras ópticas, fibras silicias.
Ujú. Sí.
What's that?
Shut off half. Because half is lighting up on you.
Okay.
This one?
Sí, sí.
Hola, hola a todas las personas que nos sintonizan desde los canales de redes sociales del Schmidt Ocean Institute. Estamos a 3.167 metros de profundidad en el afloramiento rocoso El Dorado, mar adentro de la costa pacífica de Costa Rica. Nos encantaría ver sus comentarios, preguntas, por acá los estaremos leyendo y tratando de responder.
Yeah, definitely.
Soon we will be starting our video transits, and we will do this to have a standardized measurements of the faunal composition of the seafloor. So after our dive finishes, what we will do with this data is then analyze it to see not only the species that we can find and count in the transects, but also the quantity. And this will inform us about the distribution of the biological communities.
Trying to get
How can
So this one doesn't need to be downward facing, it's only full height that's needed to be downward
this,
facing.
yeah, this.
Whereas this one does need to be as tilted down as possible. And we need the lasers on. What altitude are we at right now?
The issue that I'm having is that we need to be
Oh yeah, so I was gonna ask what altitude are we at right now? What altitude?
right now, the front of us, we're 1.5 at the middle of the ROV. We're in angles. The front of the ROV is 1.5.
Can we drop down a little bit more, if possible? Well, I think we'll avoid the height.
the depth range of the splashing is 4,500, right?
So right now it's 1.2 at the middle. That's where we're measuring from.
Okay.
So,
Yeah, so
Tenemos
we are
una
going to drop
pregunta
a little
en el
bit more
chat
just
de
'cause this is
Facebook,
flat. Well,
que cuál es la
what's that?
just 'cause this is
el rango de
this is plain
profundidad
and we're not like it.
del
Can we drop a little bit
robot
further,
SuBastian.
closer to the seafloor? This is 1.2.
Les contamos que, de acuerdo acá a lo que nos comentan los técnicos, tiene la capacidad de bajar hasta
That's-
4.500
yeah, like that,
metros.
and then just transfer just like that.
It's already off.
Yeah, but it's still
What are the settings, just so we know? Can we just write what we ended up doing?
Yes. So here. So 1 meter off the bottom.
Wait, so are we starting?
well,
Sí, los láseres. Can we have the lasers turned on?
Yeah, for sure. Without that, we'd be screwed.
Only problem is, or you can barely see them.
Oh yeah, they're there. No, they're there. They're great.
Yeah.
It's fine.
Yeah.
Um,
Yeah. So
okay, well if we're ready to go, then let's log. We just need to log start a transact
video.
whenever we start.
Just let me,
yeah.
Well,
I mean, when I'll give you the
Yeah.
signal. Yeah.
we just need to log the beginning and end of every single one.
Okay. So let's just bear with me, guys. Are we ready to
No,
start?
so
what's the speed you wanna go?
whatever-
Point zero
I mean,
three
any
knots.
randomized
Point
thing,
Or
we just didn't wanna end
point
that
0.03.
far.
So yeah, so around 60 minutes per minute.
I have it up
60.
meters per second.
Normally we just use a random number, so it's in the three, so like in the 360, so you just end up like, we can do the 45 as well.
Point
whatever you
one
want.
five.
Yeah, I'll give it a test and see if that's okay.
Hi.
Okay.
Just setting up. It's really important we write down
The speed.
settings,
Yeah.
the exact settings, altitude, so that we know. And
How's that speed?
that looks great.
Okay.
Yes.
Alright, so
That's perfect.
start whenever you love.
So this will be the speed. The
Right now we're going 0.1 meter per second.
where are you seeing that?
This is right there.
Speed over ground. Okay,
Speed
I
over
saw
ground,
it.
correct.
Yeah.
This is-
Okay. Point. Okay. So
Okay.
that kind of looks good, right?
Right,
Okay,
no,
so-
yeah, just write it on the side, so we just
Yeah.
have everything.
Yeah.
21 meters per second,
Oh, we messed up the seabed.
and then the, and then the altitude is currently what?
1 meter off.
1 meter off bottom. Right, which is that, great. And then, is there a way to like know how much zoom and tilt we have, so that we try to set it as much for tomorrow?
The tilt is gonna be it's just above, if we look down any more at all, we'll have something in the frame.
Right, so
Okay.
let's note that down.
Yep.
Okay.
As soon as the
Is there
frame's
any
clear.
specific angle you can give us,
No.
maybe? No.
No, so it's
Just-
the edge of the basket.
So
Yeah,
the
right
tracks you
Okay,
just
the edge of the
saw
basket. Okay.
were
Right
actually
there.
from Sebastian from a few minutes
straighten
ago.
out
We
via the
were kind
boards
of
right there
drifting
with that
back,
angle, and then
Okay.
it's
and now
just so
we're
that the
back
basket's
on
on.
track
That's great.
as
Okay.
we get set up for
Okay.
this
Cool.
transect.
And then zoom, just so we
There's
know.
no
So they're
zoom at
just
all.
discussing
Okay.
a
Yeah.
consistent way
Okay.
of maintaining
Yeah,
our
so now-
cameras and our lighting.
Anything else?
Can you write this
So
on the
it's consistent over all of our
yeah, on the
transects.
annotations of the video transit, like the information.
Are we starting?
I think. Are we ready to
No,
start?
no, we wanna just.
Okay. Yeah, I'll let you know.
By zooming?
I would prefer
I
not
would
to,
say-
because this is a very easy setting to reproduce. It's like edge of the basket, pulls it out. So that way it's very easy to set every time.
Yeah. Okay, when you're ready, say start a transact.
How far do you wanna go on each transit?
Yes. 100.
100 meters. Yeah.
Sorry, I'm not.
So what I'll do
sorry.
is I'll stretch this out
Yep,
100 meters.
Yeah. Okay. And then I will give you another
And this is for
random
you,
heading, and we go another 100 meters.
Okay. And we can start whenever you're ready.
Okay. So, Gus, can we start now? Our initial transit? Yeah. All right. So at
well,
48.3
the
heading.
idea of having just randomly crossing lines is that
Um.
you have a points to see that you cross the space, especially,
Yes.
and
Give me just one moment.
the idea is also we're seeing animals that might want to be sampled, so we can put like markers, whatever, to then come back. Because if we keep going in the same area, direction, we're gonna have to do ship moves, and then do ship moves back.
48.2.
So if we kind
Okay.
of do 100 meters.
So
and then
we're on, right?
100.
Okay, we're on. So
Yeah, exactly.
Yeah, we just basically right now, we're gonna start
Way
our
better.
transact bearing
Yeah.
0.0.
Yeah, that's like Mars, moon landing. Yeah, no, so you just, yeah,
I would just say,
we're here, right? You're just gonna be like heading a 48, so just go 100 meters.
yeah.
And then you stop. you
So
just-
welcome everyone who's just tuning in. We're
So you end up kind of with a.
beginning our video transects to assess the benthic fauna composition and distribution. What you see on screen is the abyssal plain in the proximity of El Dorado Outcrop offshore Costa Rica. And for those of you who wonder, we also have some two little green spots and dots on screen, and those are lasers that tell us the distance or spatial scale for us then to assess the size of things. So this scale is 10 centimeters, and again, we'll be using this to understand better the biological communities down at this site.
Yeah, it's beautiful. Yeah, 100%.
So, just talking to Diva and Jorge, that we will kind of keep the idea of doing, like, do 100 meters, reset, and then just pick a different heading.
Yes, like
Instead of
random.
going, like, in line,
Mhm.
because that way we don't have to move the ship as much.
Okay.
Like, we'll be going back over the same spots, and then if you see things, we're putting those
The waypoint.
waypoints so
Yeah.
that you could potentially come back.
Yeah.
But if we do like a long ship's move for 500 meters, then we're gonna really come all the way back.
Okay, so just random, pure random.
More or less. I mean, the concept is like you do 100 meters, and then you kind of turn around, and maybe do it like this, and then you go like that, whatever. So you're kind of like randomly
Yeah.
going over the same
Yeah.
spot. We're gonna do it 5 times.
Mm-hmm.
You know, so maybe there's another one there.
Mm-hmm.
So you're kind of crossing an area.
Yeah. Got it. All right.
We'll mow the lawn.
Yeah, mow the lawn in a very non-standard way.
Saludos a los que nos sintonizan desde Costa Rica. Buenos días. Para nosotros es un honor y un placer estar acá en la embarcación Falcor 2 del Schmidt Ocean Institute. Me acompañan 8 colegas más de Costa Rica, así que para nosotros es un honor y una gran oportunidad de estar acá representando a nuestro país y aprovechando estas oportunidades para conocer mejor sobre nuestro patrimonio natural profundo. So that will show us when we get there, right? And finish the 100 meters.
Yeah,
That will update.
down
Okay.
there.
Okay. Can we drop a waypoint here? Like a spatial mark, just to know if that, because I'm interested in those kind of them.
Okay, you have a waypoint here?
Yeah, I mean, like keep going I mean, not stopping, not zooming, just a waypoint. Yeah. Thanks.
There you go.
Okay. Thanks.
What do you want to call this?
Yeah, like let's do it in sequential order. So just waypoint 1, video transit maybe.
Start video.
Like video transit.
Marker one.
Yeah, mark 1, and let's just do it in sequential order.
He doesn't know what's the
Okay. Thanks. So guys, if you see anything, let me know, and I will tell them to drop a waypoint. I just dropped one for the starfish and the sea cucumber.
For semantic purpose that could be helpful. We'll drop markers instead of waypoints, because waypoints would change how the map acts.
Okay, so marker will be the
So
exception.
we'll drop markers for this, because
Okay.
if we drop a waypoint, then it'll change how the.
Got it. All right, perfect. Thanks for the clarification. Yeah.
I was thinking the mother first time is carcinidation.
Yeah.
They have really long legs.
So we're starting our first video transit. So we're going in a constant speed and fixed heading in order to assess the composition and distribution of the benthic community. For those who are asking in our chat, we are at an abyssal plain close to Dorado Outcrop. So what you see here is sediment that have been building for years and years at the seafloor. So it's not necessarily a sandy bottom, but sediment composed of everything that sinks from the upper water column. Usually we find here remnants of algae that live outside, residuals of animals that fall down and sink.
At least again from here.
I'm ready to ROV. Yeah, Jason, just let me know.
So at these sites, we usually have very low magnitude currents. So that's what you see here. We don't have actually a current meter on the ROV, but we can tell from previous measurements and models that the currents at this area and usually at these depths are not as strong as the currents we find in the upper 1,000 meters, which are mainly dominated by mesoscale processes and geostrophic currents. And yes, as we see on the comments, all those particles you see on screen are usually and generally called as marine snow. So this marine snow is organic matter, mainly in particles that sink down from the upper water column. And previous observations in many parts of the world have shown that the distribution of seafloor fauna correlates strongly with the amount of distribution of marine snow. So food at these depths is usually pretty scarce. So this marine snow provides very rich food for these organisms that are also adapted for different feeding strategies to take advantage of this marine snow. So many, as you can see in our screen, you have different tubes coming out from the sediment, and this is a special feeding strategy that's called suspensive feeding, and they take advantage of the particles that are suspended in the water column. But also you have the scavengers as the sea cucumbers or the sea starfish, and these are usually roaming around and feeding on the sediments constantly. So yes, pretty interesting, the relationship between the water column and how this determines the distribution on the seafloor. I have a question for you. That there, I think is pretty warm.
How far below is
The, the right one. Which one? This one down there. So we have a great question on our YouTube channel that if we dig down here, how far below can unicellular life can be found? So joining here in the control room, we have our colleague Gustavo Ramirez that is specialized in microbial ecology of the seafloor, and he is going to help us answer that question for you.
Hi there. I believe that if we dug into the sediment, we will see unicellular life, or at least prokaryotic life, that's bacteria and archaea, all the way down till we hit the bedrock. There will be no place in the sediment, or there hasn't been a place in the sediment, as far as I'm concerned, and as long as the sediment stays within 100 degrees Celsius in temperature, there is no place in the sediment where we wouldn't expect life. And actually, we would expect life in incredible densities at a minimum of 10,000 cells per cubic centimeter. If that would be a bare minimum. It can only be more than that. The average cellular load, again, for bacteria and archaea in the top 10 centimeters of the sediment tends to be in the hundreds of millions. So yeah, life just keeps on going. I think it was Jit that asked this question. Life just keeps on going unless there's something tremendously catastrophic happening, like a direct volcanic explosion or getting close to something called volcanic sills that can heat up the sediment to a temperature where it would sterilize it. But yeah, I guess to follow up on your question, if we were to dig a hole here, not sure how thick the sediment layer is, maybe 100 meters or so. If we kept going past the sediment layers into the first layer of the lithosphere, we would continue to see life, and perhaps even more there, because there would definitely be aquifers that are flowing through there, bringing in more microbes. And so the bottom line, to summarize, I guess, and make a long story short, we have yet to discover a hard line on the vertical limit of the biosphere on this planet. So the deepest hole that's been drilled, as far as I'm concerned, has penetrated kilometers of mud, and then beneath that mud, kilometers of bedrock, and all of that under thousands of meters of water, and they did not find a place without life. So that's yeah, kind of a long-winded explanation, but I hope that continues that conversation. Great questions. Thanks for asking, Jet.
Good answer, dude.
So thanks to Gustavo, and as we keep our first video transect, we see some bryozoans as well, which is the white thing we just passed. We are doing this transects to have a standardized measurement of the seafloor biological communities and even of the habitat there. So we will then take these videos afterwards and analyze it to see the distribution, and we will have several repetitions of this. This is a bryozoan we have here. The two green dots on the screen is a spatial scale, so it's 10 centimeters. This tells us the size of things. So then we can assess not only the size of the animals, but also of important features in the seafloor, such as trails or marks left by different animals that usually can burrow in the sediment.
That's really important for getting carbon sequester.
Sí, ahorita lo devuelvo a hacer otra, como que al principio lo hice en español, ahorita lo voy a decir. Es 200 metros, ¿verdad? El espesor aquí de la capa, de sedimento, más o menos. Sí. Tenemos una pregunta en el chat de Facebook de cuánto es el espesor de los sedimentos hasta el basamento basáltico. Pues con certeza, específicamente en esta zona por donde vamos pasando, pues no sabríamos decir específicamente cuánto es porque no hemos tenido las mediciones. Sin embargo, alrededor de esta zona se han recolectado observaciones que nos dicen que en promedio el espesor es de 200 a 400 metros de sedimento hasta llegar a el suelo basáltico. Y acá es donde sucede el intercambio de calor, principalmente. So we had a British stall there. Ahí está. Un saludo a todas las personas que nos sintonizan desde diferentes partes del mundo. Y bueno, especialmente a los diferentes costarricenses ticos que también nos sintonizan desde diferentes partes. Un saludo a Juan Carlos, que por ahí vemos que está. Juanca, un gran saludo hasta allá, al otro lado del mundo. Qué bueno saber que estás acá sintonizado, conectados. Y bueno, definitivamente que esto es una gran oportunidad para nosotros como científicos ticos, como mencionaba anteriormente, de entender estos ambientes profundos que usualmente no tenemos la capacidad de tener esta calidad de datos y observaciones, así que un agradecimiento especial a todas las personas que han hecho esto posible y principalmente al Schmidt Ocean Institute y su plataforma, el Falcor 2, que nos brinda esta grandiosa oportunidad de ver imágenes que de otra forma no podríamos apreciar. You can see it there, so I think we're like 70% maybe of the need. Yeah. Do you have an ETA to get like to the radius point that you?
We're going.
I mean, just to know.
Meter per second, another 30 meters to go.
Okay.
Going to
Not
right there.
Yeah.
No.
There.
Yeah.
So welcome everyone who just tuning in. We're live from Dorado Outcrop offshore Costa Rica. We're down in the abyssal plain, a little bit southwest of this feature that we call El Dorado. And for those of you who are commenting on the social media, we do have plenty of life here, the abyssal plain, as you can see now. For example, can we drop a mark here, please? So as you can see, this is full of life. Most of the time is not that easily distinguishable or to appreciate because some of these life forms can bury in the sediment. So a good proxy for us to also assess the density and the distribution of fauna is not only look for the life form itself, but also the marks and trails that they leave behind. So all those holes you see here are usually associated with a particular organism. So even though we're not seeing life, right, in the animal itself, it's probably down there buried in the sediment. And this is also pretty important for us because at the end, it plays a major role in the biogeochemistry of the sediment layer. We have a question if we're going to the outcrop on this dive. So yes, we will be heading back to the marker sites we have been studying for years and where the octopus aggregations are found. So stay tuned with us as we go to the outcrop after our assessments of the abyssal plain and the nodule fields. So we got a great question from the audience. Maybe this is for the ROV team.
Yes.
If you have used LiDAR
LiDAR?
technology, yes.
We have used LiDAR before. I don't recall us ever doing it with SuBastian, but we definitely used LiDAR before.
Okay. That's great. So as you heard from our ROV team here in the control room, there has been some LiDAR measurements, although not on board, attached to this ROV, the SuBastian. So that's a great point, and thanks for mentioning this in our comments, as we know that LiDAR is a pretty powerful tool.
Yeah. In the 4 or 5 cruises that have been on in the last 7 years, maybe used it.
Yeah. So we're almost finishing our first video transect as we see this shrimp and screen. For folks wondering, this is a video transect, so we're not doing any zoom on the specific fauna that we find because we need to have a fixed view to standardize our measurements. So bear with us because we know that you might be interested in seeing and having a closer look at all these amazing animals, but we will be doing this later on after our video transects. That was a shrimp, actually the red one that just showed. So for the person on the YouTube channel, that was a shrimp, not a fish. Son las 6:50 acá en Costa Rica. Un saludo para todos los que nos sintonizan desde Costa Rica y otras partes de Latinoamérica y el mundo. Por ahí vimos también que se conectan desde Barcelona. Así que un gran saludo a toda la comunidad hispanoparlante. Definitivamente para nosotros también es muy importante comunicar toda esta expedición en diferentes lenguajes y poder extender el impacto de estas expediciones a una audiencia más amplia. Así que, por favor, déjenos saber desde dónde nos están sintonizando, las preguntas que tienen, comentarios, por acá las estamos leyendo y para nosotros es un insumo muy valioso.
We're about 100 meters up, a little bit beyond that barrier line, just make sure that we got it 400.
Okay, so we're about to finish then. So Gus, are you ready to?
Start the transect?
Yeah, not yet. I will
Let
give
me
you
know
the
that.
mark. Yeah, but
Just think of a random number.
I just thought of
Random
another.
number.
Yeah, 77.
Right. That's the next header.
Yeah. Actually, you know what? 69.
Sure. That's where you want to go with that. Keeping a highbrow tone here this morning.
I would suggest personally, well, I guess we can come back.
Talk about
So
that.
if we're gonna go southeast, and we're gonna back down to crisscross to keep us under relatively Boston area, unless you wanna go.
Yeah, no, actually, I would agree with you, but we just been told that we want to do like really random stuff.
No, wait, so what are you saying? Sorry?
I was saying that
Go
she
southeast.
mentioned this, so we don't get too far away from where we're looking, so we can come back to look at it. So if we were to like come down like this and then kind of make
So then
star,
just
if you
choose
will.
a number between 45 and 90. Isn't that how headings work? Yeah, so that'll be in that. 77? Yeah.
Okay,
Yeah.
that's
Yeah.
good.
Alright,
So
that's.
that you, or 69 or whatever it is.
69.
Alright,
That's
Yeah.
it.
That's,
Just say the
uh,
quadrat and
yeah.
choose a random number in that quadrat.
Yeah. Okay. Got it.
Alright, so that's the end of that one.
That's the end of that one.
Yeah.
Okay.
All right. Perfect. So we just finished our first video transect.
Leave a marker here?
A marker?
Yeah. You wanna leave one? No.
Yeah, yes,
yes.
to say end of dive,
Yes.
sorry, transact one. Please.
Yeah. Thank you. Thank you for that.
Transect one.
There you go.
Okay. So you're ready to start the next one?
I'm ready to go.
Already doing? Okay. Go ahead.
Well, let me get to the proper cruising speed and then.
Yeah. Okay.
Get fast. So give me a
Are
little.
you ready to start running the initial beginning of the second one? Okay, so I will give you the mark and just write down the same settings, but it will be a different heading, right? So it will be 69.
Transect two, right?
Transit 2.
All right.
All right, so going in, starting now.
Yeah.
Perfect.
You wanna start it now?
We're gonna start it now.
Or you just gonna pass by the beginning?
Yeah, we're starting now. Okay,
Wow, we feel like, feels like we're
like
going
it's
really
going
fast
faster.
right now.
Yeah.
Yeah, I don't know. Hold on. What happened there? We'll start it again.
6 and 9.
Wait, and let's don't you, can you X out of that, because we haven't actually started it yet,
Yeah.
because we were going too quickly, so that was it.
Just put fail because I think it will. I don't know.
No,
this
just
wasn't
press
long.
cancel.
Yeah. Yeah. It was just the problem that we.
Yeah.
Uh-huh.
We have to restart.
Yeah. So yes, we have some comments in our social media channels that yeah, there might be a possibility that the animals down here are somehow attracted or disturbed from the light sources. So are we in a good speed now?
No.
No, not yet. Okay. So
Jumping from 2.
So yes, as you can imagine, all this environment that we see now lit up is usually pretty dark, to say total darkness. So it's pretty unusual for this fauna. So yes, there might be some interesting behaviors associated with light.
No, whenever you guys are set up and ready to go.
Put that marker.
Whenever you guys are, like, once you're starting to go, then we'll just tell us you're on the track and we'll draw
Yeah,
the waypoint.
and when we reach
Sorry,
the
marker.
speed, yeah.
Yeah. Just,
Alright, that's 0.1. It seems fairly steady at 0.1 down.
Okay.
correct altitude, right?
Altitude is still 1 meter.
Uh-huh, still 1 meter.
69.3.
69, perfect. Yeah.
Let's start.
Okay.
Okay.
So it's starting now. And they probably think, yeah, this is probably close to what we think an alien invasion. So
No, sorry, they're not us.
also another important thing to mention here is that even though we might seem some movement associated with our presence here, there is also normal movement of the animals in this area. So as we see on the comments, if you might see something poking up from a hole in the seafloor, it doesn't necessarily mean that it's being attracted to light, but sometimes they're just feeding and we're, just passing and seeing the behavior that they would normally do. So all those questions are to be confirmed in our post-analysis, and this is why it's such an important thing for us to have this type of transects in which we have a standardized view of the seafloor, and then we can assess many things, including composition, distribution, taxonomical purposes, and behavioral. So there's a pretty nice bryozoan. That's a bryozoan. And what we had on the right was an urchin.
Just very basic, doesn't have to be.
Un saludo a todas las personas que sintonizan esta transmisión del Schmidt Ocean Institute. Tenemos varios comentarios muy interesantes. Voy a tratar de irlos abordando uno por uno. Con respecto a la cantidad de peces que se ven, existe una posibilidad, sí, que tanto las luces como el ruido que genera el ROV aísle o ahuyente a los peces, pero también es importante comentar de que a estas profundidades, a pesar de que hay peces, usualmente encontramos una densidad menor de lo que uno podría esperar en aguas superficiales. Entonces es importante saber eso. Inclusive cuando estemos viajando a otros sitios más adelante en el buceo, vamos a ver de que asociado al afloramiento rocoso y a un sedimento más duro podemos encontrar más peces. Entonces lo que vamos viendo es que hay una repartición del nicho, como hablamos en términos biológicos y ecológicos, en donde hay especies y grupos taxonómicos que prefieren cierto tipo de fondo, ¿verdad? Y, por ejemplo, como podemos ver en estos fondos lodosos o sedimentarios, vemos muchos equinodermos eh, y muchos gusanos marinos. Y cuando vamos hacia otras partes con el sustrato más duro, vemos otros tipos de grupos taxonómicos como los pulpos o peces, por ejemplo.
No es parte de, no es parte de la cordillera volcánica submarina del Coco.
¿Quiere responder? Bueno, sí. Tenemos un comentario también muy interesante en nuestro canal de Facebook sobre la similaridad entre las comunidades en el Dorado Outcrop y la dorsal de Coco, y tenemos el honor de tener aquí en el cuarto de control al doctor Jorge Cortés de la Universidad de Costa Rica, quien es especialista en aguas profundas y específicamente que ha liderado la investigación de aguas profundas en Costa Rica. Así que nos va a estar respondiendo un poco este comentario, esta pregunta.
Esto lo que vemos, estamos viendo en estos afloramientos, en estas acumulaciones rocosas, son muy profundas, 3.000 metros, y mientras que la cordillera volcánica submarina, el Coco, la base está a 2.500, o sea, la base de esa cordillera está menos profunda que donde estamos, y las partes superiores de la cordillera están en 1.000 metros, algunos casos hasta menos de 1.000 metros. Entonces, los organismos que vemos allá o en los montes submarinos que están entre la costa y la isla del Coco son diferentes un poco diferente a lo que estamos viendo aquí.
Y ese cuento que echaste estaba muy bueno, salió en inglés.
Sí, ok. Ok, por ahí. Hey, one question. Do you have like different logs and that's going from that one? Or is it shared?
We can push them over, but we usually keep them over the screen. So
Okay. But they will at the end be on the same log. Okay. All right, thanks. So welcome everyone, just tuning in. It is 7 a.m. in the morning in Costa Rica. We are offshore the Pacific coast of our beautiful country. This is Sergio Cambronero from Universidad Nacional, and for me it's a pleasure to be joining you in this, our second dive of the Octopus Odyssey Expedition. So we were just talking in Spanish, and now I'm just going to reiterate it in English about some comments that we have in our social channels. And it's the fact that we're not seeing many fishes in this video transits. And while there might be some of the light and the noise from the ROV be disturbing and repelling some of the fishes, it is also important to note that usually in this type of habitat, which is abyssal plain, you don't find as much as fishes and as other places, for example, in shallow waters, in coral reefs, or even deep down here, but in hard substrates. So this is what we call a spatial niche segregation. So you would find that many groups have different preferences for a type of sediment or a type of seafloor. So in this, that is, as you can see, sediment and soft bottom, you would usually find common groups such as the echinoderms, which are sea cucumbers, sea stars, corals, and many sponges as well, and shrimps. And you might have some fishes here, but they're not as common as you would find in other type of substrates as hard rock substrate, such as Dorado Outcrop. And this is important because if you stay tuned, we will soon be heading up to the rocky and hard substrate where we can find another assemblage of biological communities. And in here, you will usually find more fishes and other groups such as octopus, which are the main objective of our expedition. So this is a pretty important thing to note that depending on where we are and the type of C4, that's what we would expect to see different animal groups associated. So thanks for the comment and all the questions here. So that's a pretty nice looking sea cucumber, which are very common in this type of habitat. Looks like a Banthodiotes. And as you can see, and again, as we do in a video transect, we are not zooming in. Can we drop a marker here? Put sea cucumber. As we do a video transect, we're not aiming to do any zoom on the amazing life forms we found, but we're dropping marks to come back for more detailed observations on these animals. So that's what we just saw was a sea cucumber, probably very looking to Banthodiotes. And you would tell the difference from these deep sea cucumbers from the ones in shallow water. For example, the deep ones usually are more translucent. So you actually, if you get to take a closer look, you would see the inside and the organs of the animal. While on the other hand, the more shallow ones are more fleshy. Yeah. Oh. Bryozoans. So yes, we're seeing many bryozoans in these dives. Yesterday we were able to collect one of these bryozoans, and we found that there is associated fauna with these bryozoans. Yesterday we got some skeleton shrimps, which are called coprolites. And for us, it's important also to assess the interaction between all the different animal groups at these steps. Some nice jellyfish over there. If you're wondering, the green dots you see on the screen is a spatial scale that we use to assess the size of the things we see in our video transects. So that's why we keep them on during the whole transect. And then this will be of tremendous help for us to assess biomass and other features of this region. So couldn't agree more with the comments here on our YouTube channel. This type of habitat, it's usually pretty fun habitat. You can find great creatures. Personally, I love and I'm very interested into fishes and echinoderms. So the echinoderm diversity that we see here is pretty mind-blowing. So I'm pretty happy that we're here, and hopefully later on we will be able to sample some of these creatures So yes, we've been seeing lots of brittle stars, lots of sea cucumbers, and according to literature, the echinoderms are a pretty dominant group at this abyssal and also hadal depths. We see a buried, looks like a buried sea star in our lower right corner we just passed. And it might not be that evident, but there is many organisms buried in this layer of sediment. So don't think that it's just sand or sediment. We do have some evidence of activity here, and also a good proxy we use is to look for holes or trails that are associated with the movement and behavioral of the species that live here.
I mean, someone gonna go back from this video.
So we just see a bryozoan on our left of the screen, and we have been actually seeing a lot of bryozoans. So pretty interesting group, and we're eager to know more about this bryozoans here and the associated fauna with them.
Your next transect, are you gonna come south? So heading to this marker here will be a transect, you wanna come straight from this way?
Yeah,
Actually, to how we set the ship up now.
yes, actually, I'm thinking, yeah, I think south will be good, right?
Or do you wanna kind of come back this way?
Yes, I think that will be nice. Maybe let's go 230.
230 meters?
No, 230 heading. Right, that will be that direction. Sounds good.
I think you pulled what? Gus has a random heading generator.
I have a random.
Oh, really? Okay.
What's my range? 1 in 300?
No, range will be 135 to 270.
135 to 270. Here's your number. Your random number
that.
is
100 meters at 230. So back towards
212.
Huh? 212. So that will be our new heading.
New heading from here, 212?
Yeah, from the end of the transit.
So that'd be up there.
So that marker is the end of the transit?
Yeah,
Okay.
kind of to the line that it is
Okay.
general area. So this is kind of your new.
Yeah, perfect. That's great.
Okay.
So we also been seeing many interesting animals in this dives, and there is a type of life form that we have been seeing often, and this is what we call the xenophyrophores. As you can see in the screen now, on the left, that is like some sort of rubble with texture. So that's a unicellular organism that can grow as big as a basketball. So it's pretty interesting group because there have been some association with different animals that use this xenophyrophores as a place to place their eggs and so on. So it's pretty important that we collect these video transects and have these observations to assess then the density of these specific life forms. So we see a buried brittle star in the lower right corner. And that's a pretty common behavior for brittle stars, to bury it a bit down in the sediment, and they poke their arms out. So it's a different feeding strategy to what would you find in shallow waters for this same group. So that tells us a lot about the amazing diversity and different strategies that these groups can have, and that have been shaped by evolutionary and environmental conditions. So
Yeah, ROV break.
Just get you to pause there. We're gonna finish our transect here in about 20 meters, and then we'll start heading 2 and 3 of the SOI line. Should be in position.
Okay, can make the window.
Cheers, thanks.
so we're just seeing some tubes coming out of the seafloor. These are usually associated with worms, marine worms, according to our colleagues here, experts in this type of habitats. So we have the honor to have in the control room the specialist, Dr. Diva Amon from Trinidad and Tobago, that soon will be joining you in this live stream from Dorado Outcrop. So another sea cucumber in the upper left corner, and we are seeing this, I think is some shrimps or a type of crustacean associated with the surface of this animal. So this will be pretty interesting for us to see. Have you seen this before?
What?
Those crustaceans on top of the Ventidites?
No, I saw the picture. It's so early, and I was like, what's happening over there?
Yeah, that's cool. I mean, I've seen it in three different ones. Hopefully, we get a sample. We get
And it
those
has
guys.
that, yeah.
Yeah. So I have a little cool story about sea
Question.
cucumbers. Oh,
The samples are going to where? The Zoology Museum, right?
to the Zoology Museum
At
of
Costa-
University of Costa Rica. Yeah. There is a pretty interesting fact about sea cucumbers, and even though they don't seem as fun as you could think, as other groups, there is great pieces of information that we are starting to know about these animals. So as you see, they feed on the sediment, and scientists have found that the ocean seafloor is full of viruses, and especially at this particular depths, the abyssal depths. So scientists back in 2012, I believe, there was a paper, an article coming out describing different, a particular type of symbiosis that's called endoprotective symbiosis that sea cucumbers experience with bacteria that live inside their guts. So what they found is that sea cucumbers have this symbiosis with a bacteria that has reduced and lost many of its genes in order to live in conjunction inside the gut of the sea cucumbers. But the most amazing thing is that they found that this bacteria helps the sea cucumber to defend themselves against all the viruses and bacteria that can be found in this sediment. So that's why you have probably many of these sea cucumbers thriving in this environment. So many of the bacteria and viruses, as you know, can be harmful or pathogens for animals. So based on these findings, they describe different genetic mechanisms that this bacteria use to identify and defend their host against these viruses. So the most interesting thing is that humanity is using these tools can we drop a mark here?
Yep, got it up here, that's the end, so this will be the end of
Okay.
the transect. Oh, for
Okay.
this one, yeah.
Yeah, for that one.
Sea cucumber, right?
Yes.
You wanna collect things other than sea cucumbers.
So this is us coming to the end of the transect, so I'm
Yes.
just gonna turn and try to.
Yes,
get all
and
this in.
to 200. What was the number?
212.
Yes, that one.
Is this official end?
Official end. Yeah. To finish the story, the cool thing about this is that humans are starting to use these genetic mechanisms to do genetic editing. So, based on the capability of this bacteria to identify and store the information of all the virus they have identified within their genomes. So, we find that there is not only ecological importance on this deep sea habitat, but also a really huge bioprospection and biotechnological potential. And as we like to say, most likely the medicines and healthcare advances of the future might come from the deep sea. So this is why important to not only know and understand better these environments and these animals, but also to make sure that we have environmental standards and guidelines to protect them against human activities. So we're kind of are we on a constant speed going in the heading?
Yeah, we're 2 and 2 now, yeah.
2 and 2 speed at 1 meter off the seafloor.
Yeah.
All right, so we're beginning the transit, the third one.
Yeah, I'm trying to slow down, you don't even have.
That's just cruise. No, that's right.
Just do it. Okay.
Transit 3, the same specs, right? And just the heading.
So, but you didn't even have cruise button on. No, no. Wait, don't say it. The light doesn't work, does it?
I mean, it's fine.
Yeah.
Okay, so let's just get one on this.
So for folks commenting on our social media channels, if the bathymetry of Dorado Outcrop is available, I will double check on that, and please stay tuned because I'll let you know about this. I don't have the specific answer to that, but I'll be most than happy to double check.
So this is where we're starting the transect now, just because our speed kind of increased when we came around the corner. So
Okay.
this is your point.
Okay. You start? Okay. Perfect. Cukes are really cool. We do agree with that, and especially I am really interested into sea cucumbers. So yeah, I find them amazing, and some interesting behaviors we've seen in many deep sea expeditions that they can take off the seafloor and spend some long time swimming and doing very acrobatic moves up in the water column. So we see a fish, finally our first fish of this dive down in the seafloor. Was not able to see the specific species, but we might be able to assess that better when we analyze our video. So another cuke. We have some coral bryozoans. And this is the third video transit we're doing, and this the second dive of our Octopus Odyssey Expedition.
So we may want to drop a marker here because
Okay.
one's black hole.
Okay. So can we drop a marker here? Black coral, yes. So we are seeing a beautiful black coral. I know we have some experts online, so please, we will be more than happy to read your thoughts on what this species might be. We might be coming back later to take a sample of it. Are those enough for brackets?
No, these are ophidians.
Ophidians? Okay.
Yeah, so cuspales.
Color scales, yeah. So
Generally the ugly, chunky ones.
okay. So double checking here with our expert on the control room, Dr. Diva Ayman. A lot of the fishes we're seeing are cosk eels, which are pretty common fishes at these depths. Um, so for people who are expecting fish, this is your time. Hope you're having fun enjoying the views of this abyssal plain. Son casi las 7:30 de la mañana en Costa Rica. Para todos los que nos sintonizan desde diferentes partes del mundo, hispanoparlantes, un gran saludo, especialmente para todos los ticos. Estamos buceando en nuestro segundo buceo en este sitio, el Dorado Outcrop. Y pues hemos visto cosas muy interesantes en estos buceos. Ayer estuvimos navegando alrededor de las formaciones rocosas basálticas y vimos, me atrevería a decir que entre 1000 y 2000 pulpos. Así que, can we drop another marker here?
Yeah.
For black coral?
Especially with like that weird thing.
Yeah.
Weird thing? Marker weird thing?
Marker.
We can put like marker Brisingate, marker sea star or Brisingate or whatever.
Yeah.
But so those we definitely see
And this
in the CCC.
yeah.
The purple one. Sea star. Because we'll come back and we'll sound here.
So Jeremy, yeah. Thanks for the comment, Jeremy. We know you have extensive knowledge of this group, so we might be coming back later and get some nice footage for taxonomy purposes, and hopefully we'll be able to take some samples. So please stay tuned, and thanks for tuning in. Les comentaba que ayer estuvimos buceando por el afloramiento rocoso basáltico del Dorado y tuvimos la oportunidad de ver muchísimos pulpos con diferentes comportamientos. Estaban incubando sus huevos, algunos estaban copulando, algunos estaban buscando pareja. Así que inclusive vimos algunos eclosionando de sus huevos. Así que para nosotros todas estas observaciones son muy importantes para entender más sobre la ecología y la dinámica de este lugar. Esperamos hoy, más adelante, en algunas horas, estar volviendo y regresando a este sitio del Dorado, a donde están las agregaciones de pulpos. Y lo más interesante de todo es que estas agregaciones de pulpos están alrededor de sitios en donde el agua, pues hay agua, un fluido de agua saliendo desde la corteza oceánica, que es entre 6 a 10 grados más caliente que el agua circundante. Así que parte de esta expedición, de los objetivos principales, es no solamente observar y colectar información biológica, sino también entender la hidrogeología y la hidrografía de este sitio y por qué es que estos fluidos hidrotermales están en estas zonas.
Yeah, that's what I call black girls. I was like, I know that one, and she answered it. Yeah, DV's still not. I knew the answer to that. I was
Wait,
like,
is that what?
the black corals.
Oh.
I know that one, and you answered
Do you
it.
know why it's black? That was the
Somos afortunados de tener en Costa Rica
Jeremy.
uno de 2 sitios conocidos a nivel mundial, 3, perdón, que tienen este tipo de hábitats en donde vemos los pulpos incubando sus huevos alrededor y asociados a fluidos hidrotermales. Esto hemos recolectado observaciones previas con científicos que están a bordo y algunos que nos sintonizan desde la costa, en donde se ha descrito un mecanismo recientemente y diría que relativamente nuevo para la ciencia que le llaman el sifón hidrotermal. Y esto lo que se ha visto según las observaciones físicas y geológicas es que hay zonas en el fondo marino en donde el agua se infiltra por los montes submarinos y debido al diferencial de presión que existe entre estas zonas de recarga y zonas descubiertas de sedimento, hay un flujo de agua que mientras viaja por la corteza oceánica se calienta y experimenta un intercambio de calor con la corteza basáltica y luego sale en estos afloramientos rocosos a través de grietas que están desprovistas y limpias de sedimento. Y aquí, en esas grietas, es donde se encuentran los pulpos. Ahorita mismo no lo estamos viendo, pero lo vamos a ver más adelante en nuestros buceos. So, that's a sea urchin that we have here. Can we drop a marker? Just put, yeah. Urchin and Brisingid star. Brisingid, yeah. Aproximadamente estaremos pasando y moviéndonos hacia la zona poblada de pulpos aproximadamente unas 2 horas. No tenemos un tiempo exacto, pero los invitamos a que se mantengan atentos a la transmisión. Pero por aproximadamente las próximas 2 o 3 horas estaremos moviéndonos hacia el afloramiento rocoso, donde podremos ver más pulpos. Y les cuento que para mí, siendo un biólogo marino de Costa Rica, es una experiencia increíble, no solamente estar acá viendo los buceos en vivo del ROV, sino también tener la oportunidad de verlos en vivo ya cuando el ROV sale y trae las muestras. Ayer nos quedamos hasta tarde en la noche fotografiando todos los especímenes y de verdad que es para mí un sueño hecho realidad poder describir y recolectar imágenes de estos organismos que de otra forma no tenemos la posibilidad de muestrear. Así que manténganse atentos, por favor, a todos los canales de las instituciones asociadas porque esperamos en un futuro cercano estar compartiendo más de estas fotografías que tenemos con más detalle de todos estos animales que colectamos. I want to give a special shout out to my colleagues here in the control room from the Schmidt Ocean Institute, the ROV team, which make everything, all of this possible. There is a lot of things going on behind the scenes, so you can have this awesome footage live on your computers and screens. And also, I would like to thank my colleagues Gustavo Ramirez and Tim D'Angelo that are joining me as co-leads for this dives and helping us with the annotations and observations.
So there's something on the right there that barely something on the screen.
Okay.
I was curious what that was. So we jumped the marker.
Okay, perfect. Yeah. Really, what was like?
It looked either small
Okay. All right. But it was something on the sea floor, not just. Okay.
yeah, that
Cool.
just out of view of the
Okay. Got it. Thanks. So for
What do you want to do next?
we're heading. I think we should be going right between 225 and 0, maybe. So that would be a good range, right? Yeah. So, can you throw a random between 225 and 0?
25
No, and 360, yeah, sorry.
Imprimí esto, pero bueno,
252?
252.
252. Alright.
Yeah.
So we're gonna head 252.
So welcome everyone who's recently joining us. This is our second dive of the Octopus Odyssey Expedition offshore Costa Rica. We are in our third video transect, and this is what you're seeing on your screen. So these transects we do to assess the composition of the seafloor and the distribution of the fauna here. What you see on screen are two little green dots, which are laser, and these lasers are a special scale that we use to assess the size of things. So these are 10 centimeters away from each other, and we keep this turned on during our video transects. It is important to know, for those who are wondering, that we are seeing so many awesome creatures and life forms, and we are not doing any zoom or spending time with them, not because we're not interested, on the contrary, because we want to have a consistent view of all this transect. So we are dropping marks, spatial marks, on the sites we see interesting animals, so we can come back later on and take samples and footage to identify them. So just for you to know that it's not that we're not interested, it's just that we're doing a video transect and we need to maintain the view. No.
I think it would be useful because a lot of people I think are going to come on for the octopus. So I think it might just be useful to say like we won't be getting to the octopus for like 3-4 hours.
Yeah. Actually,
Yeah.
I just said it. I don't know if I said it in English. I said it in Spanish. So
Okay.
I said it in English.
Yeah. Sorry. Just because I think like people like they
Yeah,
all
where
I
are the
see is like really
octopuses?
boring stuff.
Yeah. Oh, take a look at that. That's like a rock.
I think it's I think it's an echiuran or something that burrows in a circle and
Huh.
makes marks in
Oh.
a circle.
Okay.
We're still in the same transit.
Yeah, for another.
Yeah, we're
2 meters or
about to
something.
finish. Yeah, I mean, and I said in Spanish that we're probably getting to the octopus in probably in like 2 to 3 hours, right? I think that's a fair estimate.
Yes, exactly.
Yeah.
Perfect.
We will be transiting over the abyssal plain for the next couple of hours, and we expect to be reaching the octopus dance area in about 2 to 3 hours. So for those that are interested in seeing the octopus and the octopus eggs and hatchlings and even the baby octopuses that we saw yesterday, please bear with us. We will be getting there. We know that there are very cool observations, and we are keen to go here. We will stay for a long time in today's dive. But first, we want to have observations of this also pretty interesting habitat as the abyssal plains in this sediment, in which we find a whole different assemblage of the biological communities.
for one animal,
Oh, thanks, Dino. Thanks for the picture.
Exactly, and then people remember to switch their shoes. Or even if they just want to wear switching their shoes.
Once you turn it,
Video. No.
I can infer that just from the context.
Yeah, like,
But there's no way.
like
for me to
really
really
awesome. Like, utterly awesome.
We started the.
if you're gonna move to the west for this next move, actually.
Look at,
Did we start the?
take a
No,
look at
just,
that.
we just ended it right now. So
Yeah,
we can just
okay.
end it.
Thank you.
And-
Alright, and 255, right?
255?
252, sorry.
252, yeah. Special greetings to our colleague and lead of this expedition, Julie Huber, who's joining from offshore. Julie, thanks for tuning in. Also, that after I said about the octopus, a person put, okay, thanks, I'll be back in 2 hours.
See, like literally, people are like, no, we don't need this. I told my mom, I was like, don't come on for a few hours. I'll text you.
My mom would ask me whenever the people were piloting.
You're what?
My mom would be like, who's gonna be piloting? I'm like, so she's like, alright, I'll come in then. I'm like, appreciate
She's
it.
like in the inside track.
Yeah.
Okay, so are we,
Alright, you can start whenever you're ready.
okay, we're ready.
Alright, we're 252.1, 1 meter out.
252 we are?
Yep.
Okay, so we're going there. Okay,
And
perfect,
this
so
is 4, right?
start now, yeah,
Yeah.
Transect 4.
Alright, you wanna put a
So
start
the
point?
same one? Or 252?
Yeah, so
Alright,
we'll start
perfect, man.
Transect 4, then 100 meters out.
We're calling it video for start.
Can I see this?
Yes.
So the, oh, I see where you put in push doors.
Are these meant to be searched later?
Yeah, well, the start and stuff.
I mean, we have them on C-Log.
Okay. So I just wanna make it consistent with spaces or not spaces.
Doesn't
Doesn't
matter.
matter. I won't worry
Thank you for
about it.
thinking that
Okay.
precisely about things.
And then 100 meters
Way
out
ahead of
from here.
us.
Yeah. Yeah, but I'm on. Move it.
That's sure.
So when you're done,
No.
I'm gonna want you to go, once we switch, we'll probably have time to run 2 more of these. Like we'll probably hand over still running transects.
Yeah.
And I'll do it. So maybe when I'm running the last one,
Oh,
you go
right
in
back to
deep,
where we
and then you, if you wouldn't mind
started.
coming back.
Pretty
for the
close, yeah, that's fine.
samples, So you can tell.
Yeah.
We left there.
Did
for
you
initial
have
direction.
breakfast already?
My breakfast is just a shake, so I can just get it whenever.
Okay, alright, okay.
Your 252 pretty much to just make it right back to where we started our first transect, so good job.
Nice.
I might
You can
actually
take,
go with
art.
it now.
Yeah, totally.
Random number generator.
I mean, and, you know, like take your time.
Yeah, I'll be back.
just see that. That's crazy. Go right back to the starting point. Cool.
And then, I don't know, maybe look at due north or something like that for the last
What was that?
one. Maybe do like a due north for the last one.
Yes, yes, maybe. Yeah, yeah, that. Yeah, like
What are you gonna do?
between.
Done like this.
Yeah. Yeah, actually, but actually, it might be good to. I mean, I know it's not random, but like, if we can like cross in the middle of that, because we draw like a rhomboid,
Okay.
right? So if we can go in the middle, I think.
Just little crossing. Right.
Yeah, that will be nice. So that range will be right between 0 and 45, right? Something like that. Well, no.
About 45.
Around 45. Okay.
Maybe say last one.
Oh, okay, so I'm right with the 7 and 10 freeway.
That looks a lot better.
I'll say later.
Yeah.
Okay, cool. Greater Los Angeles or where was I?
They're by Glendale.
Orlando. Valley.
The valley. Right. There's others in
San Diego.
Yeah.
in
San Fran. Okay.
Stanford.
So down south.
Southward too.
See another one of your things.
Oh. Whoa. Sorry. Marker. A Brisingit as well, right? Yeah.
Yes, had a bit of an exodus. A lot of this. And Arizona, everybody could afford to get out there.
So many, the biggest influx of people find
Helps. A lot of people from New York, New York as well.
But you already started, right?
Yeah.
So we are in our fourth video transit of this second dive of the Octopus Odyssey Expedition. We're on top of the abyssal plains offshore the Pacific coast of Costa Rica. So we're seeing many interesting creatures. As you can see on your screen, we have a Kosk eel in the upper left corner. We've been seeing many more of them than in the initial transit. So things are getting more and more interesting as we advance in our dive. We've been already almost 2 hours down in the seafloor, and soon we will be heading to a nodule field before going into our main area of interest, which is the rocky outcrop where the brooding octopuses can be found. So yesterday we had amazing dive. We saw octopus hatching, baby swimming. We saw pretty interesting stuff. So please stay tuned as we go towards the Dorado Outcrop. So many of the rubble-like structures you see on the seafloor and the sediment are xenophytophores, which are unicellular organisms that can grow as big as a mango or as a basketball. So they're not might be that colorful, but they're definitely very interesting for us, and even more. I'm sorry that they are pretty dominant in this area. We have been seeing a lot of them, also along with sea stars, sea cucumbers, corals, and bryozoans. Those are the main groups that we've been seeing in this first video transects on the abyssal plain.
Their big thing was to go to
We see a cosquille. Oh, feeding. Okay. Someone's having breakfast. Wow. Cosquills are really common at these depths. They're usually roaming around, smelling what's buried on the upper layer of the sediment. So pretty cool to have these observations. Please leave your comments, your thoughts, your questions on the Facebook chat or YouTube channel, whatever you're tuning in from, and we will be reading all your comments, and we'll be more than happy to answer your questions.
after I got off the steamships over Chesapeake here on the tugboat for Chesapeake, over Delaware. Going from 4 months on to 4 months into.
Un gran saludo a todas las personas que nos están sintonizando desde diferentes partes del mundo en donde se habla el español. Estamos tratando no solamente de llegar a una audiencia en inglés, sino que también poder extender nuestro mensaje a la comunidad hispanohablante. Así que nos encantaría ver sus comentarios, sus preguntas, sus opiniones en nuestros canales de redes sociales, en YouTube y en Facebook. Por acá los estamos leyendo. Y para nosotros es un gran insumo saber desde dónde nos están sintonizando. All the particles you see on the top of your screen that are being lit up by the ROV light. These particles are what we used to call marine snow. So these are remnants of all the productivity in the upper water column that eventually sinks down to the ocean floor. So at these depths, food is not that abundant. So we and scientists have found over the last years that the distribution of seafloor fauna is strongly correlated with the flux of this marine snow. So in this cruise, we are not only assessing what's happening deep down, but also we're taking some measurements of the upper water column to understand better what's the interaction and the association between the water column, which is 3,000 meters deep at this point, and the influence it has on the seafloor. I don't know if you can turn off the EK80.
You made it sound like
Yes.
just some thoughts. Yeah.
Really excited. Yeah, totally. Yeah, totally. I mean, maybe present yourself again.
Are we done?
5 minutes?
No, no, we're not done.
What it is, though.
Hi everyone, this is Gustavo Ramirez here from Cal State University, Los Angeles. As we're looking at this never-ending abyssal plain here, again, as Sergio just said now and I'll reiterate, it's important to keep in mind the goal. We are studying flanks of what are known as seamounts. So these are piles of rocks that represent the crust that's protruding above the sediment layer from the first layer of the lithosphere beneath. These protrusions can be pretty big sometimes, but an interesting fact that I came across is that if you keep track of only protrusions or seamounts that are 1 kilometer or taller around the world, okay, they're very spread apart from each other, and they tend to be very small. The one that we're at is only 1 football field in size. But if you keep track of these protrusions around the world and you combine them, they add up to this. I'm going to throw this weird number out there: 30 million square kilometers worldwide. What does that mean, right? I had no idea what that meant. So I googled that, and that is the size of the continent of Africa. So this environment, the seamounts combined together worldwide are or represent a surface area of our planet that's equivalent to Africa.
Can we drop a marker here?
So again, this expedition is visiting right now the Dorado Seamount. That's the equivalent of about a football field. And this is basically the only place out of this entire habitat, as big as Africa, that we know with this level of intimacy, where geology, hydrogeology,
I'm waiting just.
and geochemistry and microbiology have been explored. So again, what we're doing here ultimately is beginning to explore a size of the planet that's equivalent to the continent of Africa, out of which we only know about a surface area equivalent to a football field. You know, at least with this level of intimacy. The geology, hydrogeology, geochemistry, microbiology of this environment. We've only studied a football field. We have the rest of Africa surface area equivalent to explore. So something to keep in mind when we're looking at this.
Thank you, guys, for that amazing piece of information. It's definitely mind-blowing. And as we are about to finish our 4th transect of this dive, I'm gonna head over and pass it to Dr. Diva Amon, who's gonna be joining us for the next 4 hours or so. Dr. Diva Amon is from Trinidad and Tobago and is the director of the NGO Species, and also an associate scientist to the Benioff Ocean Science Laboratory at UC Santa Barbara. So for me, it was a pleasure to be with you here on this dive. I hope you had a great time over the abyssal plain, and I'll be sticking around. Para todos los que nos sintonizan, agradecerles. Yo le voy a pasar el control a la doctora Diva Amon de Trinidad y Tobago. Ella es una experta en fauna de aguas profundas.
So the weird thing is a jelly.
It's a jelly. I thought it was a jelly. Así que de verdad que para mí es un honor haber estado y compartido con ustedes en estas primeras horas de la mañana. Espero que estén disfrutando este buceo. Si nos están sintonizando igual desde la mañana con un cafecito. Y bueno, los dejo de verdad con una autoridad en fauna de aguas profundas. Y bueno, que sigan disfrutando. Mi nombre es Sergio Cambronero y fue un placer. Nos vemos mañana. Voy a estar probablemente a la misma hora. Muchísimas gracias a todos los que nos acompañaron y me acompañaron a mí desde la madrugada en los comentarios de Facebook y YouTube. Big thanks.
Very nice.
While Diva's getting situated over at Science Seat, just want to welcome anyone who might be tuning in right about now. We are live from RV Falcor 2, about 3,164 meters below the surface with ROV SuBastian. Currently, we're kind of cruising around doing some transects of this abyssal plain at the base of Dorado Seamount. But we will be working our way up the seamount again today after we make a little visit to the nodule field that we saw yesterday. So as we make our way up that seamount, we expect to see many more octopus like yesterday, and we'll see what else we discover.
There we go. Thank you.
243 is
Yes.
gonna just take us just
Okay, another one.
97.
97. All
There
right.
we go.
243 is 9 degrees off of where we are now.
Which would have been fine because we just would have continued, right? But wait, so Sergio, before you go,
Yep.
so we,
How many transects?
so we, yeah, this is
This is the fourth one.
right. So we are about to end the fourth one.
Yes,
Okay,
exactly.
perfect. Thank you.
Yes, and who is Beth? Who is your?
No, Mylan. So something we're gonna raise is that like your shift begins on the hour. Please be here 10 minutes
Yeah,
before to
totally.
hand over.
Yeah, and to like update on what's happening
100%. That
yeah.
needs to like so. It'll be Myla and Miguel, and if you go, you have 2 transects. Go and eat, do all the things, and then if you don't mind and are happy to be here for their
Totally,
samples, so you can just like show what
Yeah.
you might want.
Yeah, and just for you to know that whoever is taking the annotations of the beginning initial, we're writing this on the observations and the heading.
What's SO6?
The speed over ground.
Thank you.
Yeah, SOG.
It's a G.
Sorry. Yeah, I have to test the worst typewriting. All right. All right, see you around.
Okay, perfect.
Okay, on the bridge? Yeah, go ahead. Yeah, we have a new updated waypoint for you.
Still gonna be 20 meters before I turn around.
Okay, copy on it, you guys. You can be headed.
So, good morning, everybody. Good afternoon, good evening, depending on where you're joining from. We have just had a watch change, and right now, so I am Dr. Diva Amon, as Sergio said. I'm a deep sea biologist. My affiliations are Species, which is an NGO in Trinidad and Tobago that does marine science, education, and advocacy, as well as the University of California, Santa Barbara. And I'm joined on my team by Myla Guilan, who is from I always forget the beginning of this, who is from the Research Institute for Sustainability in Germany, as well as Miguel Semedo, who's from the University of Porto, Portugal. Just to point out, on our shift, we've got 2 Portuguese speakers, as well as obviously an English speaker and a semi-Spanish speaker. So we are welcome to taking questions in all of those languages, but we may not be as proficient in Spanish as the last dive watch. So we are here, in case you're just joining us, off the coast of Costa Rica. We are just on the soft sediment on the abyssal plain to the west of Dorado Outcrop, which was the site we were diving at yesterday. And basically what we're trying to do on this cruise is many different things. We're trying to characterize what the biodiversity is of the Costa Rican deep sea, because there's been very little exploration in this area before. And we have a bunch of stuff to do at Dorado Outcrop. So Dorado Outcrop is the home to one of the two known deep sea octopus nurseries or gardens in the world. And yesterday, if you joined us, it was just an absolutely phenomenal dive. Hundreds of octopus, both mother octopus in brooding positions with eggs, male octopus, as well as we saw octopuses copulating, as well as babies hatching. So it was just an incredible dive, scenery unlike anything I've ever seen before in my time working in the deep sea. So we're hoping to get to that site in maybe 3 hours or so. And before that, what we're doing right now is we are running transects on the sedimented abyssal plain to try to characterize what lives here. We're going to be doing this for maybe sort of half an hour more. Then we're going to be doing some great biology collections. So stick around if you want to see some awesome zooms of biology. And then later we're going to be transiting to Dorado Outcrop, where we're going to be doing some more capturing some more imagery of the octopus and potentially taking some samples again. And then we're also doing a few other things on this cruise, in that we are trying to characterize the reason the octopus are, well, we're trying to understand why they're there, but we suspect it is related to the hydrothermal fluid emanating from the seafloor. There's very low temperature venting. Yesterday we got about 10 degrees and the maximum has been about 12 degrees. And so we are going to be trying to characterize what is in that fluid by taking fluid samples, as well as later in the cruise, looking at some other seamounts and outcrops around this area that have never been explored. Somewhere fluid may also be coming out, but somewhere fluid may actually be going in. So we're trying to characterize the entire environment, geology, biology, and chemistry. Okay, so you got the heading from that, right? Whatever was the one that she told you. Perfect. So whenever you're set up to do that, we are good to go. So we're logging the start and end of the transects. So when we say we're going to put in, what we've been putting in is for every single transect in comments, speed over ground.
So as we do these transects, the science team is working with our pilots here to sort of pick the direction, and we go. And if we happen to pass by anything, we're not stopping right now to sort of get a close zoom of those, but instead we're dropping down waypoint markers so that if we want to return to one of these things that we see, we can.
No, it's fine. So it's in the speed over ground, 0.1 meter per second, altitude 1 meter off bottom. The tilt of the camera is at the edge of the basket, and we're going, and then the nose zoom, and then you're going to put in the heading for what you're telling you. So sorry, that's like terrible. Once you do it once, then. And the heading this time was what? 97. So the heading this time will be 97. And so we'll tell you, transcribe. So we'll tell you when that's going to start.
Got a little sea cucumber just chilling here.
Yeah. Sorry, I thought you were the pilot. I was like, we don't need to mark another sea cucumber.
No, I was just commenting. Yeah. I like them.
They're great.
Yeah.
They're great. And one of the lovely things about the abyssal plain is that there are just so many different species down here. But no, they're great. They're like the cows of the deep sea, sort of just munching along, munching, pooping. That's what they do.
Kind of little, yeah, cows and like Roombas of the deep sea, right? They play an important environmental role of breaking down a lot of matter from other animals.
So we're just setting up to start our 5th transect of the dive. And once we do that, there's a little baby halosaur on camera, but I think that's what it looks like. And once we do that, we are then going to run the transects. We've been running them for 100 meters. They were a good opportunity to sort of get a feel for what are the main animals living out here on this sedimented abyssal plain. And then we're going to do one more after that and then begin collections. Ready? Yeah. Okay. You can put started transect, please. Here. Oh, you have the piece of paper. We continue then.
Thank you.
I made video 4 and video 5 start.
So I see there's a question about the octopus specimen and what they look like. So yesterday we were able to, very luckily and thanks to the amazing skill of the pilots, we were able to collect one male octopus and one female octopus, as well as a clutch of eggs. And whoa, that seems really quick. Yeah. In terms of whether, I mean, the baby octopus may have been one of the most cute things I've ever seen in my entire life. In terms of whether there was anything surprising about how they looked, unfortunately, the two octopus experts are not yet here. But yeah, we'll definitely come back to that question. So even though it may not look like there's a lot of animals down here, actually what we're looking at, or species, I should say, what we're looking at here is in this frame, there's probably, ones that we can see with our eyes, 30 upwards. All of these weird little marks on the seafloor are xenophyophores. These are single-celled foraminifera that create this sediment test or shell type thing that they live in. So these are most likely alive, and they are a huge part of the diversity down on abyssal plains. And then if you look really closely, you'll be able to see things like right now on screen, I can see a little ceryanthid. We can see a sea pen, an umbellula. We can see a polychaete tube worm in the distance. So here, the life is small, but it's incredibly diverse. And there's also a whole component of life that we are not seeing at all that lives within the sediment. So that is where the majority of life actually is, in the top 10 centimeters. And from research being done just west of here, we're finding that abyssal sediments in the tropical Eastern Pacific are incredibly biodiverse, some of the most biodiverse in the deep sea, which is really exciting. And down here we get, a lot of what we've been seeing today is ophiuroids. They tend to sometimes bury themselves in the sediments. You might just see the tips of their arms poking out. We've seen a lot of sea cucumbers, some sea urchins, some bryozoans, which we collected yesterday that had a bunch of different isopods living on it. And because right now, if you joined us yesterday when we started the dive, there were a lot more, what we're not sure whether they're rocks or nodules, so something hard to attach to, dotted on the seafloor. And that meant that the diversity was very different to what we're seeing now. So this is largely soft sediment, and so it means we're missing a lot of the animals that need that hard substrate to attach to, right? That are like an anchor. So things like corals, things like sponges, things like anemones. We're not seeing as many as we will in the next portion of the dive. So in the next sort of hour or so when we transit to the next further upslope before we get to the actual hard seamount substrate. So this site has been visited before. Scientists came here in 2013 and, let me start again. Dorado Outcrop has been visited before and was visited in 2013 and 2014 by scientists, but that was actually the outcrop itself where the octopus live. And they were not able to take biology samples on that. It was mostly a geology cruise. So not a lot of people that knew what exactly was going on, what they were seeing in terms of life. So this cruise is the first opportunity to come back to this remarkable octopus site that we'll be getting to a little later today. And actually to be able to sample a lot of the life down here, you need a sample in hand to be able to pin down exactly what species something is. Yes, imagery is incredibly helpful and can give us a sort of a rougher cut, and is often all we have. So it's certainly very, very critical part of doing baseline surveys, but on this cruise, we're able to collect biology, and that's really exciting for Costa Rican scientists. So just swimming to the left, that's probably an Aristeid shrimp. They have this like rhythmic, beautiful way of swimming with their pleopods, very, very large pleopods that they use, and a couple brittle stars and tubed worms. So I'm not sure what type of worms, but tubed worms. Is this number 4 or 5? Right, sorry. So this is the last one, and then we're going to begin collecting samples, and then we're going to do the same thing at the nodule. And then going up to the nodule area. So down on the abyssal plains, echinoderms are very common. We just, again, west of here, which is like what we've seen here, we see a holothurian, a brittle star crinoid, just went off screen. This is likely an nematocarcinid shrimp. They've got very, very long legs and antennae to allow them to walk very easily on these very soft sediments and to detect any prey going by. But on abyssal plains, echinoderms are some of the most common animals, largely because they are deposit feeders. So there's a lot of food here to munch through on the seafloor. And just west of here, where a lot of work has been done in an area called the Clarion-Clipperton Zone, they were finding that actually the eastern tropical Pacific is an absolute hot spot for brittle star activity. So like many more species than they were expecting, apparently very remarkable biodiversity of brittle stars. And we've certainly seen a lot of them on this dive, although we're not able to see exact species because they're often smaller or covered with sediment. Another example of an echinoderm coming into frame, this is a sea urchin. And it looks like it's got some associates living on it, maybe perhaps some crustaceans. And we're probably going to be doing some collections of these types of animals in the next sort of 20 minutes. A little jelly just floating at the top of the screen. So over the course of the next 2.5 weeks, we're going to be continuing to do dives. Apart from Dorado Outcrop, all of the other sites that we're going to be visiting will never have been visited before, so we have no idea what we're going to find. We're hoping some of them are also low temperature venting sites, and so therefore potentially also octopus nurseries. Janet Voigt, the octopus expert on board, the octopus expert, I would say globally, she deep sea octopus. She thinks that because of the numbers here, that would not be enough to sustain an entire population, and so she thinks that there probably are additional sites close by, and so we're hoping to come across some of those. But also, I mean, yesterday, as we moved up Dorado Outcrop shallower, were able to see just huge zonation going from these abyssal plains that we're looking at here. Then we went into an area with some hard substrates like nodules or rocks, we're not sure which changed the community of animals living there. And then as we progressed uphill, the substrate became harder and harder. We entered the areas with the low temperature venting where there were lots of octopus. And then just about around that area where there was less low temperature venting, just amazing corals and sponge gardens. Just as we got to the top, close to the top of that outcrop, I mean, it was just so much life in one view. Really a coral and sponge garden. So this is a really fascinating site. And I'll say it, yesterday was one of the best ROV dives I've ever been on. And so if you stick around later today, you should be able to see that range of habitats as we move through them. Can we drop a marker here by the sponge? Oh, that isn't really pretty. Well, okay, well, it's all going on, but I know Jorge is really interested in the zoanthins living on that sponge. So on this glass sponge we can see here, stalk sponge, I should say, on the base of it, it's what? Stalk sponge with zoanthid. On that sponge just about to go off screen, on the stalk, there's likely zoanthids, so another entire animal living on them. On the left, there's a worm-like looking animal, perhaps a nemertean, so not actually a worm. And then we've got a bryozoan just at the bottom of the screen, that white circular thing, some worms, that tube worm just above it, just above that, a sea pen, likely umbellula. And then just coming into screen, you can see just here how things change once you introduce hard substrate. We're suddenly getting a lot, very different life and what may seem to be like an increase in diversity and abundance, right? So we've suddenly got this big, beautiful black coral, as well as sponges living on these rocks, some other little small things that I'm struggling to see right now. But yes, you can see that really things are, to avoid knocking into it, fair enough. Yeah. So the markers we've been dropping is just, not waypoints, they're just markers that, so that we know to come back to that and to sample it, because we don't want to break the transit. So if we know to come back to samples, we're more efficient. But you don't need to note it down, because it'll be noted down when we sample, because there will be loads. Yeah. I mean, it doesn't hurt, sure, but it's not necessary. Yeah. What's up? The what? From Jerry. Oh, I answered that already. Yeah. So these xenophyophores, which are these sort of like nest-like looking things on the seafloor. They can look platy. Not that one. They'll find another one. But they're made of sediment. They're like platy or reticulate. There's two off to the left. These are really interesting in that they're single-celled foraminifera that create these houses. And here in the eastern tropical Pacific, again, it's known to be a very diverse area for these xenophyophores. Again, just west of here, for instance, there were, in the collections that were made, there were 36 species, morphospecies collected, and 34 of those were new to science and had never been seen before. And so it's this type, this is likely the same thing here because there's been so little exploration in this area. It's likely that the species that we're seeing and going to be collecting are new to science. If not new to science, they will be new records for Costa Rica. And there will be most certainly a lot that can be learned about them for their ecology. All the biological samples that are being collected on this cruise are going to the Zoology Museum of the University of Costa Rica, where taxonomists and curators are going to be able to work on them. I think that's the first stalked barnacle we've seen, if that's what that is, or maybe, or it's a serianthid. I can't see properly.
How'd the xenophyophora look like yesterday? How did it make it up?
Really good. It looked great. Yeah, really good. Like, I was surprised. The one in the core, right? Yeah. Yeah, really, really good. Looked great. The only thing that looked terrible yesterday was the sea pen. It just like literally just dropped all of its body. So it was just like skeleton and then just like all the tissue and polyps were just broke. It was, yeah.
No, the sponge as well.
Which one? The flaky one.
Yeah,
that also didn't look great. But the glass one, the Euplectella, was great, and it had the 2 shrimp living in it and everything. It was a really good specimen. So this is a sea urchin or echinoid. Probably going to be collecting one of these shortly.
So would it dare to say that echinoderms are dominating the game here, right?
Definitely in the abyssal plains, echinoderms are, if we're talking about megafauna, one of the most dominant groups, if not, for sure.
So it will be interesting to see the difference with the natural field.
So on this cruise, because many of these places have never been explored before, what we're going to be collecting is called baseline data. And it's essentially creating that foundation of knowledge, which all other knowledge collected thereafter can be weighed against. So it's a really good way to assess, for instance, change or impact. And it really is the first step in being able to understand somewhere. So while you're here, what are you thinking in terms of samples? Were there particular holothurians that you liked?
Yeah, I think that there was one that looked like a
Like that?
yeah, no. Well, there was one like that, but there was like a seabed.
Okay, do you remember which?
I think it was the second mark that we dropped. So yeah, that one and one of the bentodites,
Okay.
for sure. But yeah, I mean,
Did we label the one, the bentodites? Or did we just put all of it?
I just think that for the first one we put waypoint 1 because I thought we were putting like sequential markers and that. So I screwed up, but yeah, for the other ones I think we just put like C50.
Okay.
There was one that was called like percentage, and that was like represented in seafood.
I guess, to be honest, this whole thing is really small, so maybe if we just, like, where all the, you can see where all the markers are on that one. So maybe we just go to hit, like, the closest bunch and see what's there.
Yeah.
So we are hoping today's, I'm seeing a couple of comments in the chat that people are sad they missed yesterday's dive, but just to say that we do hope today's dive is going to be as remarkable. And we are going to be visiting a lot of the exact same spots. So do stick around. It's going to be also great. It did, yeah, I saw that. And I was like, is that from the thrusters? Yeah. I was like doing its thing. So yesterday before the dive, we were unsure, given that the last time scientists had been here was over a decade ago, the last and only time. And so we were unsure what we were going to actually find on Dorado Outcrop, whether the venting would have still been happening, whether, okay. So this is likely Relicanthus daphneae. It is related to anemones, but not actually an anemone, and it's attached to an old sponge stalk. And you may, if you look really closely, you can see the tentacles causing a shadow, and the tentacles can be like several feet long in some cases. So just west of here, we saw one that had like 6 foot long tentacles, basically. But they're just very beautiful, and were basically had their own, I think it's order created for them recently, or family. I don't want to say the wrong thing. But yeah, definitely a great one to spot. So yeah, so yesterday we were unsure what we'd find, whether the venting would still have been happening. As a result, whether the mother octopus would have still been there, whether their eggs, last time scientists were here, in the eggs, did not see any viable eggs, so eggs with developing embryos. So they weren't sure whether that was because the octopus were stressed or the conditions in the venting fluid were not good. On the contrary, it's thought that the elevated temperatures in the venting fluid could do that to help accelerate egg development, or there could be something chemically in the fluid that they benefit from. So there were a lot of questions about why the octopus were doing certain things and whether they would even still be there. And so yesterday it was, there were huge chairs during the dive. When we got to the site, saw that yes, the octopus were still there. Yes, they were still there in the huge amounts of numbers that we saw yesterday, a sea star on screen. And we were able to see octopus actually copulating, and as well as octopus hatching. So we're able to know that this is very clearly it's a nursery site, as well as just a hot spot for these moose octopus, which we, yes, did confirm yesterday. The genus is Moose octopus, but unsure of the species just yet, and whether it is potentially a new species. So some xenophyophores, sea urchins, bryozoans, and you can see there's a lot of marks all over the seafloor. So holes and tracks rocks and all types of stuff. And so all of that is what we call bioturbation, and that's where basically animals that may be feeding, burying themselves, pooing, etc., they all sort of mix up that sediment, and that's one of the big processes that helps to actually sequester carbon and lock it away for geological timescales, and basically is one of the big reasons that the ocean is one of our biggest climate allies. It's because of what happens down here on the deep sea floor with carbon being locked away as a result of many of the animals that live down here's actions. Great, thank you. So that's the end of our transect. So now we are going to do, so now we're going to do some collections imagery and collections of animals. So if we could head over to where we drop some of those markers.
So we finished up with that transect, and now Diva's working with the pilots here to organize some collections. And as I mentioned, as we were doing those transects, as we passed over items of interest, animals of interest, or areas of interest, we would drop markers. So now we're actually able to use those markers and go back and selectively collect some of these articles of interest.
Yeah, so thanks for zooming in. Great. So maybe we could go back to, let's go to one of the first ones, the species, the sponge with zoanthids.
All right.
We'll grab that one, and then we can grab the, oh my god, my eyes are like failing. What do I want to say? V? Video.
That's the video. It says coral, spongy video start video.
Let's just start with the sponge with the zoanthids.
All right, so we'll go back to that one.
And then when we get there, we'll do some really close-up video, both with the lasers on and lasers off, and then collect.
All right.
Yes.
So I was thinking that for the CP conversion, let's just try to have to collect the ones that have the demonstrations on top.
Do you remember which one I want?
No, I don't know. I mean, let's see. And I don't know if there's still.
Why is it on yours?
I saw them also on some sea urchins as well, interestingly.
Yeah, that'll be pretty
Um,
interesting to see. So
So
yeah,
maybe one holothurian, one urchin, or,
I mean, yeah, for sure. Yeah,
we haven't seen any of these, so we'll leave that till the nodule field.
and the
we can
Brisingate
do-
was pretty interesting too. The Brisingate
Oh,
star.
okay, so the Brisingate close to here,
Is
so we'll
it?
go to that. So would you prefer the Brisingate to
Instead
the
of the
urchin?
sea urchin.
Okay.
Yeah, although, yeah, I don't know.
So we'll do, here we'll do one black coral, one holothurian, one Brisingate, and a sponge that has zoanthids. And
Okay.
then we'll not do any more collections until we get to the nodule area, because that has like quite different.
Okay.
I think we'll see like more black corals, more
Or,
corals.
yeah, totally. Yeah.
What did I just say? Oh, the sponge.
Yeah, even, for example, maybe if we're not doing like the sea urchins, at least we can, like stop by and like get a nice footage. Maybe people will be able to identify it with, just the
Yeah,
video.
I think. Oh, good shout actually. Yes, that's a good idea. We can
Like
go
a
and
video sample.
get great imagery without, or we can collect some. No, let's just go and get some.
What does that mean?
holes are the sheets, the power sheets that push and maintain the tension on the we'll don't have to. We can leave them on, but they make a lot of noise, I guess.
So, as Alex said, we dropped a bunch of markers, and so right now we're heading over to one where we saw a beautiful glass stalk sponge, which has zoanthids living on the stalk itself. And so we're going to go over to get some footage of that, as well as then do a collection. We're also hoping to do collect a Brazingid sea star, one holothurian, and one of the black corals that we've seen this morning as well, before we move on to heading over to our next waypoint, where we're going to do some other sampling and transects.
Yeah, I'll just toss up the map here real briefly, so you can kind of see what it looks like where we have all our little waypoints laid out there. And you can see the ROV, the large, I don't know, magenta rectangle in the corner. That's actually Falcor 2. And so we're just making our way around to these various waypoints, at least the ones of interest.
Those are markers, not waypoints.
Excuse me. Yes, I've been corrected on the terminology. Markers,
And
not
then
waypoints.
If we zoom out, then you'll see the waypoints.
Ah, perfect. There we go. Yesterday I was used to
So the
just
waypoints
like the
look
3
like
waypoints that we had. Yeah. Sweet. Thank you.
Yeah, we're confusing things. Oh, so this was where we saw the, I was like, oh, sponge, and you were like, but look at the coral, right? Yeah. Do we know if Wendy or Oda are awake?
Yes. I don't know Wendy, but Oda is.
Can we ask whether she wants the whole thing or a snip?
I'm gonna go double check that.
And we'll do the sponge in the meantime.
Zoom in on seems a bit far away from that coral. That's okay. Just retrace my line.
That's okay, we've seen other one, yeah. We'll
I'll retrace my line.
It was kind of moving to our starboard as we were
Yeah.
going east.
Ooh, there's something big in front of us.
Yeah, I don't remember seeing that when we did the
We're gonna go check
line.
that out regardless.
Yeah.
Is that it?
Oh, isn't,
Is that it?
I think that, yeah, that's the big, right, so that's the big black coral with, that you were like, oh, look, the coral. So we're just gonna, before, we are coming back to sample this one, but we're just gonna double check what exactly is needed. In the meantime, sample a sponge, which is close by. I mean, that's a giant black coral, so I'm gonna assume she does not want the whole thing, or we'll look for a smaller specimen.
I think we can get a bit of that.
So someone's asking how we decide what samples to collect in terms of the biology. Well, so for instance, this morning here, last night we discussed amongst us what are the priority samples. So for instance, on this expedition and right now, some of the priorities were black corals, octocorals, sea cucumbers, and some sponges. And that is largely because those are the expertises that exist within at the University of Costa Rica's museum. And so two of those, so for instance, we have a black coral expert on board, we have an octocoral expert on board, and we have someone who has a keen interest in holothurians and is well on his way to being a holothurian expert. So, those were our priority items. As you would have seen yesterday, we are also collecting some octopus because that's a key part of this expedition, and we have an octopus expert on board. So yeah, so before we collect, if we could just get some great footage of with lasers on and lasers off. But things that, apart from that, other things that guide our sample collection is whether something might appear to be new or remarkably strange, or perhaps if we're characterizing an area and it's one of the most common animals there, and so something that we will need to know what it is, we would do that. So yesterday we collected a sea pen because it was very common, obviously the octopus. So yeah, there are lots of different factors to take into account. Other things that we consider is, how many might there be, because we always want to sample sensitively, how easy the collection might be and whether we have the tools on board the ROV to do it, and so on. Great. Is that max zoom?
No, it's not.
Yeah.
If I go zoom, the
Yeah.
glitches go away. It's like-
Okay, so that's perfect with the lasers, and then if we get some great footage close up. Yeah, so you can see this sponge. This is a glass sponge, and you can see its stalk is actually made up of like individual silica strands, basically, that look almost like spaghetti, right? They're covered in sediment, so they're very dirty. But when we pull them up, they almost look like a fiber optic cable, kind of, with lots of different strands making it up. Beautiful. Oh, and how'd that go?
It's pretty awesome.
Yeah. Can we just tilt down slightly so we can just get the base? So the whole thing in screen. Yeah, perfect. And you can see that this sponge stalk here has been colonized by these zoanthids. So these are not anemones, they are colonial zoanthids, and they are not part of the sponge. They are a separate animal living on this sponge. Okay, that looks great to me. Maybe let's just do the collection. Which I think if you think it would fit in a coral quiver, let's drop it in the coral quiver.
All right, so
Thank you.
the porch out and then you
So
can
getting ready to sample. Yeah, no. We're not doing it just yet, but we will. Just telling you.
pick up the,
We're planning on collecting that.
Yes, for the zoanthids. So,
And it's probably.
sure, but so things that we're gonna try to get 4 around here before we go to the nodular area, because in the nodules there's different ones, more octocorals. So we're gonna try for a black coral, a brazingid, this, and a holothurian. Question about the black coral. Do you know, is a snip enough, or does she need the whole thing?
Depends on the size. If
'Cause
it's
the-
like that,
The whole thing.
the whole thing.
But if
Yeah,
it's
the whole fence is
big,
it. But if it's,
if there is a big one close to here, and we were like, it would be irresponsible to collect the whole thing.
oh yeah,
So do you know if any snip works? Like just a snip of one of the side
but if it's gonna be
branches?
collected with the by suction.
No.
we cut it off with the jaw, put it into all the curves.
Can you pull it?
Wait, not, what are we talking, the sponge or the black coral?
Oh, we'll take all this.
Yeah, we're gonna take all of that and just put it into a quiver.
Some of that, Chris?
Wait,
Yeah,
what's
that's fine.
happening right now?
I just didn't, I'm gonna put the porch out, I didn't wanna run that over. 'Cause it was on
Run
the
what
seabed.
over?
The handle.
Oh, sorry. I was like, why are we gonna suction? Sorry, yes, got ya.
That's
Yeah,
what
no, so we're gonna pick up the whole thing and put it in.
Yeah, those could have, some of those have rods that are
The
big,
sponges,
this one.
sorry,
So let's see.
just like, yeah.
Sure.
So if we, when you're collecting this one, try to pull, instead of pulling towards, pull up.
From the very.
Yeah, from the base, yeah,
Dig down a
from
little bit
this,
to-
from the stalk, pull up, because it might extend further down. Well, definitely does, and so we want to try to get as much of it as possible. Please. I asked on Jorge, but we're gonna try to collect a black coral after this, and so it's quite a big one. So I just wanted to make sure, now that you're here, like how much we should spend.
So I was telling that what
So
You just take very, very close-ups and very good pictures, and you can collect just a piece in the action.
So we'll show you after, because we're gonna go there straight after. Big, like a meter.
Like a meter, yeah.
So-
Yeah.
So we'll just get some good imagery.
Good images also of the
Base.
podcast,
Yes.
close-up of the podcast, and then at the down part.
Yes, we did with lasers on, beautiful, like beautiful close-ups.
Lasers and no lasers.
Yes, exactly.
And collect a piece in the.
Wow.
For science, it saves automatically in C-Log. So every time you do one, that saves. And so.
Yeah, you could see all the fibers hanging off that.
But also we can go back to anywhere in the video. Yeah. Beautiful sample, guys. So here we're getting like 2 for 1, right? So that's a great reason. Here you can see the actual, we can see the silica strands there. So here we're getting 2 for 1. So there's a keen interest in sponges on board and a keen interest in cnidarians, which the zoanthids are. And because it's actually 2 specimens, not just 1. Perfect. Good work, guys. That was great. Okay, so now we're gonna try, once this is covered up and we're good to go, we're gonna go over to the big black coral and get some great imagery of that, please. So to continue answering Okan's question, not only is this a 2 for 1, but it's also, and there's an interest in it, but also it's likely because so little work has been done in this area, both of those species are probably new. So once back on shore, the experts will work on that sample and basically look at its morphology, so how it looks, as well as probably its DNA. So do molecular work on it, and from that they're able to create a whole picture of it, write out a description, give it a name, and then it will formally become a species
So Diva,
or a known species.
when you have we've got limited spaces on the porch basically.
Yeah.
When you find one of those kind of 2 for 1 or 3 for 1, is that bumped up kind of in the targets then?
Definitely, because it certainly means that instead of having to use each of our limited compartments on the ROV tool tray, instead we're getting 3 into 1. So certainly that bumps it up.
Cool.
Yes. Was that in coral quiver number 1?
101,
Yes. Was the one. So if you put into the text, put like sponge with zoanthids, ZOANTHID. Perfect. No, thank you. So we're gonna head to the black coral now, because that's like just right here, just behind us. Then we're gonna do a holothurian and a brazingid. And then we're gonna move to the area with the nodules. Brazingid, sea star, the ones that have like the big, yeah. No, not nopheroid. It's a sea star. It looks very similar to nopheroid, but is bigger and has like the suspension feed. And then because we want more octocorals and black corals, we'll save the rest of the samples for the nodule area, so we can get.
So in another day that we'll go back to the abyssal plane, pick up the seriatulids.
The cereanthids, okay. Or, I mean, we can scratch the brazingid. Would you prefer a cereanthid? Up to you.
Either of them,
Okay.
whatever. Oh, yeah, that's nice.
Yeah, so it's a big one. So we'll get some good imagery of it. And then, Oda, if you would tell us, like, where to snip when we get there. Not now, just in, like, 5 minutes. Yes. Are we close to waypoint 2 right now? What's the closest waypoint to where we are?
Dive start, nodule field,
Well, they don't have numbers.
marker,
Oh, right, yeah, so done, so we're close to waypoint 1. Yeah, thank you.
So we pretty much have done 100 meters from that.
Just in like, what, sticking in the neighborhood. So we're looking at an absolutely beautiful black coral. You can see from the lasers shining on it right now that are 10 centimeters apart. This is a pretty big animal. And so we're gonna get some great imagery of it, both with the lasers on and off. So we're able to do measurements, but then also have some great footage without the interference of the lasers. And then we're gonna take a subsample of it. So looking at it now, roughly, where, like, just maybe if 2 or 3 of those.
Cut
Yeah.
from the top
Oh, I see. Okay, so like,
of the piece that,
So go right in the right.
Like, so, like
in the center
here.
part and then cut it just-
yeah,
yes, I think so.
Okay.
I think both ramifications.
okay, sure.
But it is important to go close to the holdfast
And get imagery.
before the holdfast, just in case to notice the polyps, if they are polyps or not.
Okay.
In the holdfast.
So once we're settled, we'll do some really close zooms and some big outputs. We settled? Yeah, so if we could do, if we could do zoom in a little bit and then keep lasers on just to get some quick shots. And then tilt down a bit, if that's okay. Right, so, and then if we could do some lasers off very quickly.
Some glamour shots.
Some glam shots, exactly. Beautiful. Beautiful. So that's gorge. That's perfect. And so then if we could zoom even more and then work our way from the holdfast up the body, just getting some close.
Polyps in the holdfast, you see, there are some species that do not have that. That's
Okay.
why this is important.
Can we zoom any more? So if we could just zoom a little bit more on the holdfast itself and focus on the stalk. Yeah. Yes, that's great. Perfect. That's great.
You can put the laser to have the idea.
Might not be able to see the lasers at this.
Oh, because we're zoomed in too much. But we'll zoom out and get some with the, yeah.
Okay, so yeah, I'll zoom, get this as reference, I'll zoom out.
That's beautiful. So we're zooming in certain parts because we need to look at certain parts of the animal to be able to identify it. Yeah, so then let's do some zooms of the branches as well.
There's that, get that lasers on there to get an idea.
Oh, see? Yeah. So that whole fast is like
It looks like this, maybe.
the. Oh my God, gorgeous. Gorgeous. Yeah. And so you can see very clearly now why black corals are black corals, because their skeleton underneath that tissue is actually black. Okay, and then if we could just zoom out a bit and do just work our way up. Yeah. Take a look at the commensal. Is it? I think it's a wormy thing. To be curled so tightly, that's weird.
Can we go closer? No. It's the closest that you can go.
Unless I can move up closer.
No, I don't. Yeah, in case we kick up a bunch of sediment, that's not. And then if we zoom out a bit and keep working our way up. Get some, great shots of here what maybe look at what the white thing is off to the right. There was a little white thing like there. Yeah, perfect. This is great. And so Oda, sorry, if we could hold it there for a second. So Oda, you think like from here
Yes, I think so.
up? Okay,
Maybe up a piece.
perfect.
So you see where this is
So beautiful.
right in this area? Yeah.
So black corals are actually are some of the oldest animals on the planet. There have been very few deep sea animals aged, but there have been black corals close to Hawaii that have been aged at over individual animals at over 4,000 years old. So we know very little about what age this might be, but given how slow growing many of them tend to be, this could be a very old animal. Which is amazing when you think about like old growth trees on land are something that's so special, right? Whether it's the sequoia or whatever, we consider them to be very special parts of our culture and our lives. And so it's interesting to think that some of these are as old, if not much older than those. And you can see that this black coral is this three-dimensional structure that is home to lots of different animals. We're seeing something, I think a polychaete maybe, shuffling along there. We saw some other types of animals. But we're just getting some really great imagery because this is going to be essential given we're not collecting the whole animal because of the reasons I just mentioned, the fact that it is very old, it is very large. We are going to get all this great footage so that the taxonomists can use it to then work on descriptions if it does need to be described with the tiny sample we're going to collect. This is beautiful. That bit's not looking so good. Sure.
So where those wrapped around?
Sure. So, and then there's a little crustacean. So if we could just go back to the tightly curled thing, that, and just zoom as much as we can. I mean, I don't think it's an ophiuroid. I think maybe it's like it's too, it's wrapped so tightly. It looks like literally like yarn. It's not, obviously, but maybe it thinks it's
Yeah, we've
an
seen,
ophiuroid.
we've seen that on Octopus.
Yeah, I've seen it on the curls too.
I mean, sure, you see them, but never wrapped this
Not
tightly.
this type, for sure.
Of course, octocorals live on these things, but that's really tight. Okay, so if we can zoom out again, just get another whole shot of the animal. And
So we're going to cut that up there, but down here, the half of the animal is too much.
and then I think whenever you guys are ready, we can sample, like where we pointed out. And if you think it can fit into a quiver, pop it into a quiver again.
If he gets where we're looking at, shoot.
Great, perfect.
The only issue, my lights are a little bit messed up right now.
Also, I think because we're at the beginning of the dive, and we have so much to accomplish. Let's not do that right now.
Still a great shot.
I think is this yours?
Nope.
Is this yours? The cameras? Oh, okay. Fine.
Okay.
These are yours, though, right? What's up? Just don't tell me. Yeah.
Whoa, you gotta move it anyway. Alright. We're gonna have to move it. Well, I can come around at different angles.
Yeah. Can we just get a quick? Are we able to zoom on that like mucusy thing?
Oh, port arm? Start up.
And
It'd be pretty
the-
slick if we had a little switch or something.
And the forearm doesn't have the blades on it, the damage if you pick apart, so that's much it messes with it.
And the port arm has intermeshing claw, so that like,
I am not sure what this is.
Oh, we can
so
bring it with
it's
us if you want.
easier to pick up.
No, it's fine. I just I was just interested. Okay, let's get the.
So they
Or
just
maybe
do
like
that.
Italian relic cantus?
No, it's weirdly curly.
Very fair. Se parece con relic cantus, pero muerto.
He's moving it out of the way.
Or how? Don't worry.
To take it off first?
No, we can't open the tool tray without
Damaging it
Yes,
without hurting
yeah.
that wood if they're on there, we don't wanna do that, so.
For next time, would it have been easier to reposition the vehicle or to move the rock?
See how difficult the rock is first.
So next time, let's do that.
How long?
Because I think that's also better. Maybe let's just reposition the rock,
yeah,
reposition
we're
the
gonna fly on the
ROV.
other side.
Yes.
The current is kind of coming
Back,
directly at us.
going
which
that
is
way.
convenient.
What I'll do is I'll just,
Just go on that side.
I'll probably just move over to the right, like.
Oh yeah, so the thing, so the current, so it takes the current away. Yes.
Should still be able to get that, no problem.
But I think they should be fun, yeah, hypothetically, yeah, it just kind of depends on the situation.
Maybe not. It's just the rough one. A second.
Like kind of buried in the sediment. I would say so. Maybe that's sure some activity, but I haven't seen that occurred. There might be. That's my best guess.
Should still be able to reach over there or not?
I'll do a test read
Okay.
first, I'm not gonna cut anything.
Put that, yeah.
Oh, yeah. There's probably a room in there. Right?
Yeah, well, I'll be able to bring the portion so you be able to go down farther. Should be able to. Just get the
Yeah,
top
the-
off and then we'll have,
That's
you're
plenty.
behind it, yeah, you're good.
Okay.
So we're just setting up to take a snip of this black coral because it is quite a large individual, nearly a meter, and as a result, probably quite old. So we're going 2, right? Coral cover 2, right? Ooh.
Alright, I was gonna put the fortune, but I don't know if I trust myself to do that. That's also not. If you need me to, better reach or something.
Okay, I guess.
Yeah.
Like smashing something on it.
If we could get like 5 or 6 branches down. Yeah, between 5 and 7 branches down, 5 and 8. Perfect. Is this the one with the blade?
Yeah, but they're tough sometimes. So.
It is flexible. The problem is that maybe you have to cut it.
Just don't pull it, just rotate.
Or maybe reopen and clamp again.
Rotate just a little bit towards
So there's a question out there while we're doing this. Do black corals have immune systems? Can they, for instance, die of infections or disease?
There are not much diseases reported for them,
Okay,
but they have natural protection.
right.
That's why they have been used for medical products, for pharmaceuticals.
Okay. So with regard to the question about whether black corals have immune systems and just generally corals, whether they have immune systems, yeah, they do have various mechanisms for protection. And in fact, some of those are, because of course they are like animals, but we really don't know very much about the ecology and especially sort of the biomedical, I guess is one way to put it. Yeah, the biomedical aspects of it. And so, but interestingly, something amazing about the deep sea, and particularly with, for instance, corals, oh my gosh, it's not even barely cut, is that because these organisms live under very extreme conditions, and because of the diversity of life down here, there's now, or there has been for a while, but it's increasing. We're now looking to the deep sea more and more as a source of marine genetic resources. So essentially, many of these animals contain compounds, whether they're enzymes, whether they are bacteria, things we can learn about their immune systems, for instance, that can help us potentially in life. So, for instance, some of the great examples I use about this when I'm speaking to the public is that new sources of antibiotics might come from the deep sea, given antibiotic resistance is one of the major challenges to face humanity in the future. The anglerfish, for instance, has this amazing reproductive mechanism where the male anglerfish actually fuses to the side of the female anglerfish. And because it's able to do that and her body doesn't reject him, there's a lot that we're learning there about immune responses and how to prevent new organs that are being put into humans when they have organ transplants from being rejected. And then, for instance, the glass sponge that we collected earlier, other glass sponges are being known for, are being used as a sort of a template to essentially develop new and more efficient fiber optic cables. So there's a lot of solutions that we might get from the deep sea in the future, and this might be a sustainable way of using deep sea life in the future. We don't want to detach it from the rock. Suggestions.
Well, if we held it with the other manipulator, like.
And pull, like, tear. Yeah, let's do that.
To give some resistance.
So just carrying on from what we're talking about, some of the applications of the properties of some of these animals, genetic properties of these animals. I see there's mention of a cancer drug from a sponge. So yes, that is true. It's from a shallow water marine sponge, and it is one of the most effective treatments against breast cancer. So it's a great example of how the ocean potentially, we're able to use it to benefit us in a sustainable way. Good work, guys.
No rush now.
Pardon?
Don't want to lose it now.
And yeah, someone's raised another shallow water example, the horseshoe crabs, and the fact that we're able to get some of the compounds that are able to show contamination in medical environments comes from horseshoe crabs and their blood. So yes, that's another great example. And given the extreme conditions in the deep sea, it's likely that there's even more things down here that can help us. We just because we're only just exploring these places, we haven't really been able to tap into this amazing reservoir of potential solutions. So as you saw, because black corals, this is a very large black coral, potentially very old, we took a very small piece of it, and we're going to be able to use that to potentially describe this new species by using the imagery as well as a sample to look at how it looks, its different features, as well as its DNA. So, and then it's going to be housed in the Zoology Museum at the University of Costa Rica, where local scientists will be able to use it and global scientists will be able to request to use it as well. So this sample, we try to sample really minimally and have as little impact as possible. And these samples are used for basically the rest of their lives and will help to inform how to potentially protect these places in the future. That was great, guys. Thank you. So now we're going to go to the Brazingid, get that. And then we're going to go to one of the holothurians and get that. And then we're going to move.
We're going to move more than 100 meters away.
Yes. Actually cover some ground.
Is that close all the way?
It should be all right.
So this is a, thank you, Joanna, for your comment. This is a great time to remind us that, you know, the pilots here are absolutely exceptional. And their skill combined with the dexterity of the arms of the ROV mean that we are able to collect everything from rock samples to taking snips of corals to, wrangling octopus, to even getting temperature samples by holding things into the fluid as well as fluid samples. So really we are able to do so much thanks to the skill and the amazing technology on board. Do you want to say that in Spanish? We don't have to. But you're right there. I was like, what's happening?
Hola Joana y a todos los que nos ponen sus comentarios en redes. Pues sí, definitivamente, como dijo Diva, la habilidad que se toma y que se requiere para este tipo de muestreos, pues son muy meticulosas las habilidades que hay que tener para no afectar al espécimen. En este caso, pues no queríamos colectarlo todo porque era muy grande, así que pues acá estábamos igual respirando profundo y esperando de que todo saliera bien, y pues inclusive tuvimos que utilizar los dos brazos, pero por dicha el entrenamiento que lleva el equipo técnico del ROV es bastante bueno y se logró. Así que bueno, saludos igualmente a todos los colegas de El Salvador y de Centroamérica.
Thank you, Sergio. That was great. So now we're heading over to where we dropped a marker earlier, where we saw a special kind of sea star called a Brazingid sea star. These are, well, I'll explain it when we see it, because then it'll be easier to understand. A sea star. So we haven't seen any yet. So as soon as we see one, we are going to. Yeah. Are we moving in direction of the purple thing on the seafloor? Is that close to, just because it's, I think, a weird thing, but not that I want to sample it, just to see it close up, but if it's a wave from the person, we don't have to. Okay. So while we are on our way to find a sea star we saw earlier, I just wanted to get a quick glimpse of this animal that's coming into the center of the screen. So this one is I remember the first time I saw it, it was just west of here, and we were totally confused by what it was. I mean, looking at it now, you're like, is it an anemone? Is it a sea cucumber? But actually, when you zoom in, it's actually a jelly that lives on the seafloor turned upside down. So if we're able to get some good zooms, we'll be able to see that these are actually, the jellies are essentially upside down, and these are its tentacles sticking up. And it's just a weird thing. Yeah, so there you can see much more clearly the bell or Medusa was sitting on the seafloor. And then you can see it's sort of inverted and its tentacles are facing upwards. I'm not sure what the white parts are, but yeah, just weird. That's, can we just flick the lasers on to get a sense of size? That's okay. Even if it's like now, whatever, but just so. And so this is, and so just some, I mean, what we think is, we don't think this is something's wrong with or anything. We think this is just how this jelly lives. And there's a similar jelly, again, I'm not sure how closely related they are, but there's another type of jelly that lives in mangrove forests, so in the shallow waters, mangrove lagoons, and that jelly also is inverted, sitting on the seafloor like this. Weird, right? Yeah. Yeah, sure, go for it. So one of the chief scientists, Jorge Cortez, from the University of Costa Rica, is just going to say that in Spanish.
Aquí estamos viendo una anémona de mar, una medusa, muy interesante, porque las medusas normalmente están nadando en la columna de agua y hay algunas que viven sobre el fondo. Y las que conocemos bien son las casiopeas, que son en las zonas poco profundas y están como vueltas hacia arriba, con la boca hacia arriba, y tienen algas simbiontes. Y hemos encontrado esta en aguas profundas aquí en Costa Rica, que igual vive viendo hacia arriba, probablemente para capturar alimentos. Y ahora podemos verlo con los láser, que está de 10 centímetros de un lado a otro, que es lo que mide más o menos esta medusa.
At the next site. Thanks. Cool. That's cool.
It is about 10 centimeters.
About 10 centimeters.
Great. Okay. We can continue on to the Brazingid.
You think they're upside down to just receive the food? Because then with the Cassiopeias, because of
Right,
the,
Cassiopeia, that's the genus. I couldn't remember it. Yeah.
Because of the symbiosis, but yeah, we've seen those before.
These jellies.
In these waters.
So they occur in the CCZ. That's where I've seen them, and we didn't have this good imagery, and so we were like, is that an anemone? Is that? And it wasn't until like several cruises in that we were able to get such good footage that we were able to see it was a jelly.
So we'll come up, spin around, and fly back along the trail and see if we can find it.
Yes, please. That would be great. Hopefully they're not moving. It wasn't moving too quickly. Is that it? Right? No. Ooh. Oh, so there's one of the weird pagurids. Some more black corals, ophiuroid just going off screen. Swimming sea cucumber.
Not on the line we were on earlier. It's not like this.
Well, after we can go and get that purple sea cucumber in the distance. Either of those. Since we're right here.
There.
Yeah.
You move the vessel due north. Just like move the vessel up 20 meters due north.
Sergio, are
Hey,
you
on the
okay
bridge,
with this
we
one?
get
We're
a vessel
approaching.
move due north at 20 meters.
Yeah, maybe not.
Roger that. Thank you.
Let's try another one that has the shrimps.
Oh, we can't see if it does or doesn't, so let's get a good look quick.
Looks like none, but yes. If that all three is the one we want. The thing is that we saw.
I'm pretty sure I'm seeing little white things on it from here, but we'll get a good zoom. Yes, Sergio.
All right, let's go for it.
So we're going to collect this sea cucumber we're looking at here. We think it might be in the genus Benthodites, but I'm not sure. We think you can see some tiny little white flecks on it, and those may be commensal living crustaceans of some kind. We're going to get a closer look to see what it is, but we're going to try to collect all of that again. So it's like a bonus sample, right? We're getting 2 species for the price of 1. Can you just pan down for a second, or tilt down? Sorry. There was just something weird moving. Oh, see, it's like a little worm. Don't worry. There. That's fine. Okay. Does this have? Okay, there was another one close by that we just saw. We'll go and check that one, but if it doesn't have them, we're going to collect anyway, just in the interest of time.
Yeah, that's right.
So if you wouldn't mind, just going to the other sea cucumber we'd just seen, I think it was just there.
So beautiful.
I should have gotten some better image, it's fine.
Yeah, but that's really cool because if they left, if they were here and they left, and then it's like they come and go, right? Yeah. It's pretty interesting.
I mean, it's like well known that there are other ones that like hang around. We say like ride the bus to get somewhere quicker, right?
Yeah.
Like polychaetes and other things. So the other one was just to the left somewhere, we think. Yeah, great. Yes, please. And this one does look like it has one of the little things on it.
Collecting one more time.
So if we,
If I listen gently,
oh, and it just
and it
went through. Yeah.
And it was like this big to that big. This is my boy. I was like,
So holothurians, yeah, they're largely composed of gut. You'll be sometimes be able to see that gut through them translucently. That's a big part of their body. And there are some species, the ones that are particularly able to like swim, whether for short periods or longer periods, and those often before they swim will do a big poop to basically help them lift off and swim more easily before they do that. Oh, so it's got a little mysid or shrimp or something on it. Okay, that's great imagery. If we could do some with the lasers on and off, and then collect.
May I see your dive plan?
Sure. Oh, sorry. Do you want the whole thing?
Yeah. Thank you. I just wanted to.
Oh wait, can I show you something? So, because not all of these animals were in this area, when we get to this area with the boat closed, we've saved some of the samples to do here. That's okay.
That's great.
Did we get the lasers on with this one? Oh yeah, perfect. Okay, so yes, we can collect whenever you're ready.
all right, what do you want to put?
What do you suggest?
I mean, a coil clipper would work,
Sure.
and it would fit, and
me
once
see
it
the
closes, you're
plan.
not gonna worry about it.
Scoop these up, yeah.
Are these bio boxes sealed?
Oh,
I think let's just go with a quiver.
Yeah, there's stuff in
Yeah.
all the boxes.
Yeah, let's go with the quiver.
You need to pour it?
yeah.
All right.
Number 3, I suppose.
You work for me?
Actually,
That are you do any of them really? What's your oyster?
just debating on which one we need easier.
My only concern with going too far to the starboard is that you will be coming across the bow much more. Which may be a problem, might not. Yeah.
So there's a great question about why have color when you live in total darkness. Well, there's a couple reasons for that. So one is that it's not total darkness. So in the deep sea, we scientists over the last few years have realized that probably about 70% of animals in the deep sea around there are able to create their own light or bioluminesce. And so we imagine that when we're not down here with our bright white lights, that this is an area where there is still a lot of light, just no sunlight. So bioluminescence could be one of the most common forms of communication on the planet. So there is some light down here. The other thing is that pigmentation, just like it could be a product of something they eat or something that's come from their diet, potentially. But for others, where it's more structural, that could be as a result of their essentially wanting to, just like on land, right? They may want to camouflage a bit better. And so we know as you go deeper in the water column, not only does light decrease, but different wavelengths of light, because light is made up of different colors, white light is made up of different colors, different wavelengths, those different colors will disappear more quickly. So the first color to always disappear is red. And so that means that you cannot see red light in the deep sea. And so as a result, anything that is red or purple, like the sea cucumber, will essentially appear to be completely camouflaged or invisible. So it's like an invisibility cloak, if you will. And that's why so many animals in the deep sea are often red or purple. And we can see in the background one of these. Oh, there are two actually in shot right. Oh, three in shot right now. 3 different xenophyophores. These are these single-celled foraminifera that form these tests. And you can see they've got 2 different morphotypes that we're looking at now. The sort of plate-like one, platey type one on the
back, and then a more reticulate one. Great. Oh, it's right there on the bio box. It's on top of the bio box. Yeah.
Hold on, Sebastian. Grab it. No. We have three options. We try to dig down there and grab them and leave them, or I can fly up and then fly down to see if he floats up and then grab him out of the water.
I like that third option.
Same.
Less destruction of
Yeah.
the rest of the equipment.
Yes.
There is one question in Spanish about the pressure change. So how do they make how do we achieve that the change in the pressure doesn't affect the size?
Okay, so I can answer in English, then you answer in German. So I see there's a question in Spanish about how the pressure change as we bring these animals to the surface might affect these animals, and whether we're able to prevent it affecting the animals. Unfortunately not. If in order to keep the conditions of the deep sea as we move up to the surface, we would need to have a pressurized container, which is very technologically difficult. So it means that the pressure and the decreasing pressure and the increasing temperature does impact the specimens as we work our way up. Yes, they are in these various coral quivers or bioboxes where there is water, and they are sort of semi-sealed, so they do stay colder than the surrounding seawater, but they do not remain at the temperature of the deep sea. So unfortunately, by the time many of these animals, they do go through a very big change in pressure and a very big change in temperature as we bring them to the surface. And that usually results in them arriving at the surface not alive.
I've done that with the bio box before. Yeah. And
Como bien decía Diva, vemos un comentario que mientras estamos muestreando el pepino, que se nos fue un poco al lado del recipiente de muestreo, por ahí preguntan y comentan en el chat que cómo se hace para que la presión, el cambio en la presión no afecte las muestras. Entonces, desafortunadamente, no se puede lograr como eliminar ese efecto que puede tener el cambio en la presión y en las condiciones, porque habría que tener una cámara presurizada que en este momento no contamos.
Absolute legend.
all right, you got him? I'm gonna come back down.
Paréntesis porque logramos capturar el pepino. Entonces, como venía comentando, habría que tener una cámara presurizada que en este momento no tenemos. Entonces, todas las muestras van a, pues, a experimentar un cambio no solamente asociado a la presión, sino también al cambio en la concentración de oxígeno, en la temperatura y en la salinidad. Sin embargo, específicamente respecto a la presión, sí les podemos decir que se minimiza el impacto para evitar el barotrauma, que es cuando hay un cambio muy abrupto en la presión y cuando, por ejemplo, se saca algo muy rápido y se le salen los ojos y se le salen los órganos. A veces eso sucede mucho en la pesca. Esto no pasa con este tipo de muestras porque el ascenso y el cambio en la presión es muy progresivo y lento. Entonces, al menos el aspecto de la presión se trata de reducir para evitar el barotrauma. Entonces, muchísimas gracias por la pregunta y el comentario. Y bueno, ahí seguimos leyéndolos.
Thank you, Sergio. Thanks, Sergio. So we're just going to stow this guy, or girl, or both, and take a quick look for the brazingid. And if we don't see it, I suspect the next site we're going to, there'll be many brazingids that we can collect. So we're going to head over shortly to a new site, or to a new waypoint, I should say, in this site. Yeah. I mean, that could be a pelagic. I don't think it's there.
Yeah.
That's okay.
He's like, I'm off.
Yeah. There was another one close by, so you just probably went to that one.
Yeah. That was incredible how you catch it in the air. Did you see that comment? This is the day you almost got it.
Except we did.
Hey,
Yeah.
Alright, this is right.
Yeah, it's pooped a bit from.
Scared the
We want to sample that for
Go in. Yay, well done. Great.
Amazing.
So I think let's actually just head over to waypoint 2.
Okay.
And then when we get over there, we have a bunch of different things to do.
Is that right? Just said, it said station escape.
And if when we're transiting there, we just don't go too, we'd like to keep the seafloor in sight and just in case we see anything interesting.
It might be easier to get
So we are just stowing the sample, and then we are going to head over to our next waypoint, which is going to take us closer to Dorado Outcrop, and where we begin to move slightly upslope. And once we're there, we're going to do a bunch of different sampling.
Yeah. A little bit more on the backside. That might be easier. Yeah.
Largely we're going to do some rock sampling, we're going to do some water sampling, and we're going to deploy some traps for microbes. And then we're going to do some more biology sampling as well as 3 more transects. So we've got a lot to do at the next site. And then after that, that's when we're going to head up to Octopus City, or the Octopus Garden, we should call it, or nursery. So we're probably still a few hours away from the octopus, if that's what you're sticking around for. But I mean, there's lots of other great things to see down here, and the site that we're heading to is probably going to have a bit more to see. Thank you. That was great, guys. Really appreciate it. Good morning. So we're just in the process of doing an RV pilot change, and then we're going to be on our way. Okay, settled?
Yeah.
Okay, so we're gonna head to waypoint 2, please. And if during that transit, we could just keep the seafloor inside in case we see anything, interesting. But it doesn't have to be like transect setup. Just, yeah. So in case you're just joining us, we have just had an ROV watch change. My name is Dr. Diva Amon. I'm a deep sea biologist and based at both Species and NGO in Trinidad and Tobago in the Caribbean, as well as the University of California, Santa Barbara. And on this shift, we are joined by Miguel Semedo from the University of Porto in Portugal, as well as by Myla Guilon, who is joining us from the Research Institute for Sustainability in Germany. So in terms of the actual watch shift right now, we have an English speaker and 2 Portuguese speakers, so feel free to throw over questions in those languages. But we also have a lot of Spanish speakers in the room, even though they're not officially on shift. So we can certainly try to tackle any Spanish questions as well. So we are just to the west of Dorado Outcrop, which is, if you joined us yesterday, where we saw some absolutely amazing geology, but more importantly, biology. I mean, hundreds of octopus all in one area. It was one of the most remarkable dives I've ever had in my entire career. Certainly, I mean, the scientists on board the Falcor 2 were just absolutely over the moon last night. So we are going to be, we're going to be heading to waypoint 2, where we're going to do some fluids, some rock biology samples, some more transects, and then we're going to head up to Dorado Outcrop. And the point of this whole expedition is essentially to try to learn about the entire deep sea ecosystem off of Costa Rica in this area. This, apart from Dorado Outcrop, where we dived yesterday and are diving today, the other sites we're going to be visiting have never been visited. Dorado Outcrop has only been visited on 2 prior expeditions. So these places are very poorly known or not known at all. And so we're trying to understand the geology, the chemistry, and the biology so that we can basically understand what this ecosystem is like, and then use that to inform, for instance, management of Costa Rica's waters, perhaps through future protected areas, or if, for instance, they were to understand human impacts in these areas. But yes, this is critical information we are collecting for Costa Rica and also the surrounding regional ocean and global ocean. There's a fish, probably an ophidiid or cusk eel on screen right now. We just saw an aristeid shrimp swim by. So one of the other things that we're doing on this cruise, apart from gathering that critical baseline information, is that we're trying to understand, more about this remarkable octopus nursery at Dorado Outcrop. So there are only 2 known octopus gardens or octopus nurseries in the deep sea that are known so far, one off California and this one in Costa Rica. And so we're trying to understand why this is such an important site. Why do the octopus brood their eggs here? And potentially, given the numbers we see at this site, we think there may be other sites close by. And so we're going to be looking for more sites in the future. One of the reasons that we suspect the octopus are at Dorado Outcrop is because it's slowly emanating low temperature vent fluid. So yesterday we got temperatures of about 12 degrees from the fluid, between 10 and 12 degrees. And the ambient water of the deep sea is about 2 degrees. So that is a big change for deep sea animals. And we think these octopus may be using that in some way, whether it's the chemicals in the fluid or the increased temperature to help their eggs develop or to potentially help themselves. So we're trying to understand that relationship. And we're also going to be looking at the microbial relationships with the fluid and the seafloor, because that in itself has a lot to do with the whole entire functioning of the ecosystem. We're going to be looking for other areas where fluid might be emanating from the seafloor, hydrothermal fluid. And importantly, we're going to be looking for sites where if something is coming out, it has to be going in somewhere, right? So we're going to be looking for some of the sites, particularly one site called Tengo Sed Seamount we're going to be visiting in the coming days, where we think it may actually be the recharge site for this fluid. So Dorado is the out discharge, where the fluid is coming out, and we think that Tengo Sed may be the recharge, so where the fluid is going in. And there's only one other place in the world where this type of relationship has been, observed before. So these are really critical observations and measurements and samples being taken that are going to have, again, very far-reaching implications. Was that from us? Yeah. So, in case you're wondering, we have this is a brand new ship. I think we're the third shakedown cruise for the Falcor 2. The ship is absolutely remarkable. I think the scientists are over the moon to be on it. This is, I think, Cyclopodes sempariana, or longicorda. It's a sea cucumber that has a big sail. It's laying flat right now, the big sail, but it does have a super cool sail. And there's a couple different species of sea cucumber, 3 just were within shot. And so the scientists are, like myself, are just over the moon to be on this absolutely incredible ship. On board, there are about 60 people in total. The science party is about, I think, 20, and 8 RV pilots. And essentially, we work in shifts of 4 hours, the scientists at least, and then beyond that, we need to be available when the ROV comes on deck. So the ship does run 24 hours a day, and we try to split our tasks so that there are always people on board. But it does mean sometimes a lot of early mornings, a lot of late nights, basically because we just never know, or we think the schedule is constantly changing, so we need to be on call for that. So last night, the ROV came on deck, I think at about 7 p.m., and scientists, we were all awake until about 10, 11 to process all the samples that came on board. And then the RV went back in at 5 this morning. So some of us were awake at that time. Well, hit the sea floor at 5 this morning. And so some of us were awake at that time and have been, and are going to cycle through the day. So you're going to hear lots of different voices. But just to say, we are incredibly busy, and we've just had one dive, but it has been a phenomenal expedition so far, and we cannot wait to see what the next 2.5 weeks are going to uncover. So as we're transitioning the slope, what we might begin to see, and what we saw yesterday, is we're going to see some zonation. So here right now, we're on a very heavily sedimented abyssal seafloor, but we are moving up slope. And so what we're probably going to see is begin to see more of more hard substrate coming into the picture. So yesterday we saw some rocks on the seafloor that we were calling nodules, thinking they were polymetallic nodules because they were covered in this metallic crust. But actually, they're not that at all. We made a mistake. They are, in fact, lavas, so lobate lavas, pieces of lava that have then become encrusted in this polymetallic crust. So we're probably going to begin to see more of those coming into sort of sitting on the seafloor, and that is going to create a much more mixed environment, not just soft sediment, also hard substrate, and that's going to result in a different set of animals coming into basically the community. So we're heading over there now to do various types of sampling, and we're also going to do some biological sampling there in addition to the rocks and water because of the change in species, species in the community. So we can see some of them coming into play already. These are these lobate lavas covered in the crust. And ahead of us, it looks like there was once a very large coral attached to one of these, but over time it's died, or yeah, it's not dead yet. It doesn't look like it's totally dead yet, but it's definitely not doing well. And that may have just been that it got too big actually for the nodule, or sorry, not the nodule, for the lobate lava, and the whole thing sort of keeled over, and then it's not able to live very well in the sediment because it relies on the current for food. So as we're transitioning on, it doesn't look like there's a lot down here at first glance. This is a classic thing with the abyss, the abyssal zone. But actually, when a lot of the animals here are small, either small or smaller, or actually live within the sediment. So there is a lot happening from the xenophyrophores. We can see on the sediment surface this Brazingid sea star here, sea cucumber ahead of us. But also when you look very closely, there's a lot more down there. So we'll begin to see little bryozoans, little corals, little anemones. It really does, in these environments, it does take sort of stopping and looking in a very focused way so that you begin to understand how much there actually is down there. Thank you. What's the last one? I'm sorry. Did you get one? Oh, thank you. So just in the center of the screen right now, that's a sea pen from the genus Umbellula. We can also see some sea cucumbers in the distance. I'm not sure what that pink is. Oh, a Brazingid, don't worry, it's fine. A bryozoan that's about to go off screen. We collected one of those yesterday, and it was home to, I think, 18 or more skeleton shrimp or isopods. So I'll caveat answering this question by saying I am not a geologist, I'm a biologist, but so nodules, the ones that we get, for instance, in the Clarion-Clipperton zone west of here, those form in a very specific way. So they form like a pearl where there is a central nucleus, so something like a grain of sand or a shark's tooth, and then over millions of years, this metallic crust accumulates on it. What we're seeing here is different. So what's likely happened is we started with these sort of lava rocks, so lobate lavas, and then those have become encrusted with this polymetallic crust. So the polymetallic crust in this case is likely very thin compared to the nodules, which are only essentially that polymetallic makeup. Yeah, this is great. Yeah. So are we going to MKR? No, where are we going? No, we're going to the blue.
Yeah.
Got
Yeah,
ya.
I'm just gonna come up
Thank
with
you.
section line and just on that direction.
I'll answer later. It's fine. Yes.
Someone's asking where you're from.
Okay. So on the current Science Dive Watch, I'm Dr. Diva Amon. I am from the Caribbean, Trinidad and Tobago to be specific, and I have two affiliations. The first is an NGO that I co-founded called Species, which does marine science, marine education, and marine advocacy, as well as the University of California, Santa Barbara, specifically the Benioff Ocean Science Lab. And I'm joined on this shift by Dr. Miguel Semedo, who is from the University of Porto in Portugal, as well as Myla Guilon, who is joining us from the Research Institute for Sustainability in Germany. But in addition to the three of us, we have a room full of excited scientists from the US, Canada, and most importantly, Costa Rica. More than half of the science party are Costa Rican scientists, which is really important given we're working in Costa Rican waters. And then we've also got, of course, our RV pilots and our, what's the correct term for you? No.
So I'm multimedia technician.
Right. We've also got Alex, our multimedia technician, and then Steph, our marine technician in the room. So it's a full house, and this is how it's been for all of the dives so far. Everyone is really excited to see what's happening. Even though there are screens all over the ship, you just don't get the excitement that you get, that you get in the control room. Can we just get a quick zoom on this urchin? Because that's the first time we've seen this kind. So far we've seen some green sea urchins, but this is the first time we've seen one like this. And I'm just going to pull up my... So this might be some kind of like Eurochinus or something. But as you can see, it's very different color to the ones we've been seeing before. Reddish brown, and is not quite as like conical isn't quite the right term, but yeah, that's great. Thank you. We can carry on. And you can see it's left this trail behind it as it's moved along the seafloor. Who knows how long it took just to walk that short distance. Can we get a quick zoom on that? Like stick-like thing? Sorry. This. This? It's there. It's here, yeah. I didn't walk with it. Didn't bring it in my bag. 1 second. Oh yeah, we could just carry on. Thank you. Let's continue. Yeah.
Hola, un saludo a todos los que se conectan desde Costa Rica y otros países hispanohablantes. Mi nombre es Sergio Cambronero y quería aprovechar para enviarles un gran saludo a todos los que se conectan en este momento. Vamos de camino al Dorado Outcrop y pronto vamos a estar viendo muchísimos pulpos y demás animales. Así que, bueno, pues me da mucha felicidad ver a varios conocidos por ahí en el chat, así que manténganse en sintonía. Pura vida.
Oh. So was that a squidworm? Maybe. Don't even know. It's fine. Yeah, so Kat's just said that she loves having scientific experts and partners from the areas that are being studied. This is something that's been really long overdue in the deep sea science community, particularly, and it's something that Schmidt Ocean Institute is working really hard to actually improve in the future. So they certainly are leaders in that respect, and it's really exciting to be a part of this amazing expedition with so many local experts and regional and global experts. Great sea cucumber here and brittle star, probably just having a chat about what's going on this Sunday morning. I don't know. But we actually haven't seen this sea cucumber yet, this white one. Perhaps a Molpadiodimus or something like that, or Mesothuria. Yeah, I don't know. Another one of the green sea urchins. This is great. Yeah, we can just, if we just continue and then just get quick zooms as we're going, exactly as we've been doing, that'd be wonderful. Please.
The nodule that they grabbed yesterday, did that come up relatively intact?
So it's not a nodule, it's an encrusted lobate lava. We've been corrected, but it did.
Okay.
And it had a little coral on it, bonus coral from NOAD. And yes, it's somewhere around, but we can pull it out. Sure, we can pull it later. Yeah, no, it was really good condition, which is an indication that shrimpy, that the polymetallic nodules west of here are really crumbly.
Okay.
Whereas this one was like pretty good to go. So that fish just about to go off screen is Ipnops meati, and it's got the reflective bit on the top of its head. It's eye structures. Oh, can we get a zoom on that thing? That one? What's this? Do you know what it is, Oda?
I love when the scientists like suddenly get up out of their chairs.
I think it's a tube, a worm tube. Because if we zoom on the end, I think the worm is sticking out the end of it, and then it's just been colonized by something. Yeah, so
It looks like
interesting. Looks like.
not like an empty tube, maybe that.
No, so I think we can see, you can just see the white of the worm's head sticking out of it. Maybe. Or that's a little crustacean hanging out. But the worm might be in there, but it could still have other stuff living in it. That's great. We can just continue. Thank you. What? Don't worry, we're going to the octocorals. We're going to the octocorals to get more octocorals. I saved 5 samples so that we could get more stuff. Yeah.
Not sure if you have explained before what are the spiral things that you usually see on the floor.
So as we're driving along the abyssal seafloor here, you can see there's a lot of different marks on the seafloor, from like holes to little burrows to occasionally these weird spirals. And so all of this is what we collectively call bioturbation. And this is one of the main ways in which it's basically all as a result of animal activity. So it could be animals that are burrowing, animals that are feeding, animals that are pooing. And all of this helps to sequester carbon and lock it away for geological timescales, so thousands to millions of years. In particular, we've been seeing some that are like a coil, and that's actually leftover holothurian poop. So as you can see, as we're coming into this area with some of these lobate lavas, we're seeing brazingids, we're seeing little corals start to be here, we're seeing ophiuroids, we're seeing a lot of different species than we've seen early in the dive, because we're now getting these animals that actually require the hard substrate to attach to. So as we continue moving towards waypoint 2, which is where more of these lobate lavas are found, we're going to begin to see much more of this and then do some sampling. Great, thanks. So there's a question about whether that tube that we were just looking at, the sort of brown tube, we've seen a lot of them. If you keep your eyes peeled, you'll see more of them as we're driving along. Whether they're cibellid worm tubes, which is a type of polychaetes from the Cibellidae family, and they're some of the ones that have that feather duster at the end of them. And actually, no, they're not cibellid tubes. We got a glimpse of the worm living within them, I think yesterday, and they do not have the feeding sort of plume. So we're not sure what it is, but it is not a cibellid. So this sea star, well, it doesn't even, potentially looks like there's a black coral, which looks like a big feather plume coming in screen, but there's a white animal to the left of it that may look like a coral to many, but it's actually, in fact, a sea star. And it's not a brittle star, it's a sea star. And it's called a brazingid sea star. And you can see it's got these sort of, first it's hauled itself up off the seafloor onto like a sponge stalk or coral stalk left over. And that's allowed it to get up into the current. And it gets up into the current because on each of its arms, you can see it's got those projections coming off the arms. And those that uses to strain any water going by so that it can get food, basically. And so the higher up into the water column and away from the seafloor, the stronger the currents are going to be and the more food there's going to be. And just to the left of that, you can see one of those polychaete tubes. And to the right of it, a stalked crinoid. So that's great. Thank you. We can just continue going to waypoint 2. So suddenly we're entering this field of lobate, encrusted lobate lavas, right? So suddenly we're seeing the habitat, the community, the animal community changing. And suddenly we're seeing these very delicate white corals. We collected one of these yesterday. They're a tiny species of primnoid coral or octocoral. Some black corals, 2 were just within frame. And a lot more of the animals that rely on that hard substrate. Also got a fish joining us, probably a cuskeel. And once we get a little closer to waypoint 2, we're gonna do a few more biology samples and then rock samples and water samples as well. Yes. Yes. So you're unless you want to go to bed, you'll be the last thing at this next site. So I think so where we're heading, nodule field. Oh, perfect. We're nearly there.
Perfect, yeah.
So I think we can do some
Yeah,
get the
take your time, I have no rush.
nodule. Okay, fine. Thanks, Gus.
I like that there's a differentiation of the salt field and the non-field.
Yeah. So so apparently no nodules.
Got it.
We were wrong yesterday. Little jelly going by, lots more corals. And so that mix of soft sediment and hard lavas results in lots of different niches for animals to actually live in. So there's less competition. So it means that there can be more diversity present. Oh, swimming sea cucumber, maybe penny agony leander, floating by. Where's Oda? Do you know where she went? Oda. Okay, I'll wait till she comes back then. No, it's okay. We're also beginning to see some sea stars. 3 in shot right now. Lots of corals. Wait. So. So we're just zoomed in too much to see them. So waypoint 2 is down here. And then nodule field is waypoint 2. So it's like, so I don't know if you have the sheet.
Yeah.
So it's waypoint. So waypoint 2 is that nodule field dot, which you can see here. So they've just named it differently today. Yeah. I was also confused by that too. Yes.
Can we see that we are already at the waypoint 2 or on the way?
I would. And how far to waypoint 2?
About 20, 30 meters.
Yeah. So we are nearly at waypoint 2. We're about 20 meters away. And we are, now that Oda is back in the room, do you remember which corals we collected yesterday.
Yes, sir.
Yeah, I don't want to collect the
You
same
collected
ones.
two bamboo corals, yes, I remember. Don't have to collect it different.
Yeah.
For example, we haven't collected this.
That one. You don't think that's a bamboo coral? Could be one of those.
Maybe, but it's different.
Okay.
And the others that are like California, something like that.
Right. Okay, a big one. Okay.
Some of them, maybe some branches.
So the Calagorgia will need to wait till we get onto the. Okay, perfect. Can we get a quick zoom on this one? This coral? This one? So this might be this. I don't want to chance it. And next thing, this is the same one we collected yesterday.
We don't need that, we collected that.
We collected that. Okay, perfect. We can just continue
But we
these
can take
pilots.
some
Oh yeah. Okay.
to have the idea. Amount. We can get some video instead of just a sample, but it's important to have the video.
Yes, you're totally right. Do we think that's the same one?
That one, yeah, I think it's
Okay, so when we get to waypoint 2, the first thing we're going to do is we're going to collect one of these rocks, please. Yes.
What is this orangey?
So if we can get a quick zoom, I think it's a different type of sea cucumber that we haven't seen yet. Just a quick zoom. Yeah.
Okay, cool.
So there's two species actually there, right next to each other, different
What is
species.
the worm or?
No, both sea cucumbers.
You can see there, squeak,
You can see what?
there.
No, good. Yeah. Oh, you said excreta. You used the proper term. Oh, wait, who's doing the push towards? That's you the push towards?
Yeah.
Okay, and so did you want, I forgot about that, did you want, you said you wanted in between nodules, but
Yeah.
maybe one or two with nodules.
Yeah, that would be fine too. So we just
Well, you
wanna-
tell us what you want,
Yeah.
That it's
and it's
not,
8, right?
It's gonna be really hard to,
but it's not,
yeah, not
aggregates.
aggregates.
But it's the
Wait, what?
big
Lava
word.
rocks.
It's not-
Sorry, I keep saying sorry. I keep saying nodules, but it is, yes,
No.
lava lobates, lobate lava.
Couple of things. We just, yesterday we collected from the abyssal plain, so it's no rocks around whatsoever.
Right.
This time we want to collect here, but there might be a ton of rocks under the sediment, so we're not sure of a push course even
I don't,
gonna be that smooth. But-
okay,
but let's
shall we
just give it a
try?
Give it
Yeah.
a shot. As soon as we know, we know.
Okay, so first we'll collect a lobate lava for microbio, and then we'll do your push course.
Good.
That's okay.
I'm gonna do that right now.
Yeah.
And then after that, is it bio traps that can just come out of the rocks?
Sure, and then we can do the
And then you
Niskins.
guys can
Yeah,
move on.
and do the
Bio traps
bios.
open
Yeah,
up space for more bio samples.
sounds great. Thank you, Gus.
Kind of soft up there.
So that, no, I think that one's actually an animal mound. So there's some kind of echeuron or something in there, and that's what's created that structure and with all the lobate lavas around it.
So this is the nodule.
Well, so now we're calling it lava, lobate lava.
Okay.
Yeah. So this is waypoint 2, and this is great. And so if it's okay with you, we'd like to collect one of these lobate lava, as well as wherever we sit down, we're gonna try to get 8 push cores from there as well. You want them all in one?
Yes.
You want them all in one spot, right?
Close to each other as possible.
Perfect.
That you won't be able to get too many because it's pretty hard underneath the set. Let's try it.
Yeah, so I think if it doesn't work, it doesn't work, but. So if it's too hard, do you want to go back down?
We need to waste ROV time.
Do you need the samples?
We do.
Yeah. Well, so then, we'll just go down to more sedimented area. We'll test it first when we collect these on
Let's go test first.
and then we'll just head back slope a bit.
Before we go too much deeper up the hill.
So once we've collected the lobate, we're going to try to do a core. If it doesn't work because of what you said, if we could drop back down slope a bit, please. So while we're setting up to collect one of these rocks, there's a question about how the samples handle the pressure differences. In terms of, I'll answer from a biological perspective. So in terms of the animals, it's a huge change in pressure and a huge change in temperature, going from 2 degrees to like 28, 30 degrees, and then going from very extreme pressures to much less pressure. And unfortunately, on the way up, that usually kills the animal because of the change. It's really hard to keep them in a contained environment on the way up. It would require a pressurized container, which is very technologically challenging. So we don't have any of those. Instead, when they're in the coral quivers or the bioboxes, they are able to be kept a bit more cool because they're semi-sealed from the surrounding seawater, but it's not completely sealed. So, and then once they get on board, we put them straight into the cold room to prevent them from essentially rotting or degrading. And then we take photographs of everything and then begin to take snips potentially for DNA or for microbiome, and then preserve all of the specimens. Yeah, I don't think that's not mine, so it's yours now. So we actually collected one of these lobate lavas yesterday, and it also had one of these little corals on it, octocorals on it. So we now can confirm that these little white corals that you're seeing here that are very numerous, those are primnoids, so a family of octocoral primnoidae. And they're super delicate and super beautiful. So this one needs to go into biobox 1. Yeah. Yeah, please. And you can see it's going to be labeled as a rock. Perfect.
This is good for you,
In terms of what? You're gonna go for this one? Yeah, sounds great. So this is a great example of what we were talking about earlier, where you try to get more than one sample for the price of one. So here we're getting a rock sample for geology. And it's also going to be used for studies of the microbiome, so the microbes living on it. And then additionally, it's got this beautiful little primnoid on it. So this is actually 3 samples in one. Great. So that's okay. So while we are, so while this is a geological sample, standing behind me is Costa Rican octocoral expert Odalisca Bridi, and she's very excited to be getting yet another sample. So I see we're joined online by Professor Les Watling from the University of Hawaii. And Les is pointing out that these corals, these primnoids, do look like ones that occur west of here in the Clarion-Clipperton zone, where there are, unlike these lobate lavas, there are polymetallic nodules over there. And yes, these do, Les, we were talking about that yesterday, these look a lot like many of the primnoids. And it's not just been the primnoids, there seem to be some species, a lot of species reminiscent of those we see in the CCZ. So it's definitely one of the things that we're going to analyze after this.
Choice of side?
I don't think we're supposed to have a rock.
Well, no, so this one says collect one lobate lava sample from microbiology, stow in biobox 1, record as rock sample.
All right.
So as long as this is biobox 1,
We'll survive, that's okay.
yeah, that's, it doesn't matter, wherever's easiest for you.
You don't need a picture before I put it in.
No, it's fine, because we dropped it, we got, yeah.
I want my rocks remaining sterile.
Oh,
That's okay.
wait, so why, so did we know that they were in, can we pause for a second? Did we know that they're in biobox 1, your stuff?
Where's the sequence for my stuff?
Oh, yours are down here.
Okay, I think my experiment
We can
will
put this,
survive.
no, we can put it back down right on the seafloor, take out your stuff, drop them here, and then,
Yeah, that's
yeah?
okay. Can that be
Sorry,
done?
pilots, apologies.
Sorry.
If we could just plop that back on the ground, we're coming back for it,
We're not going anywhere.
and we're going to pull these experiments out of this box, lay them on the ground, deploy them, and then put that into the, apologies about that.
I guess I can give
Thanks.
people the rationale, it's just, in those PVC cutouts, there's some
Wait,
sterile.
do you want to say? So we're just in the process of switching the order we're doing things, and that's for a very specific region. So I'm going to hand you over to someone to explain that. Gus.
Hi, everyone. So there's ton of interesting biogeochemistry that's happening at this site. So one of the things that I'm interested in as the microbial ecologist, one of the microbial ecologists in this expedition
So
is...
we can begin to pull out. Can we do more than one at once?
Yeah, you can bring them all at once. And,
All the yellow
as long as they all
ropey
stay
things.
within about a square meter, that should work. Meter. So, okay, so the experiments that we're about to deploy on the seafloor here are basically 5 different types of sterile substrate materials. Kind of like the first order question that we had coming out here is what drives the microbes that come out of fluids to colonize different rocks? Is it temperature? Is it simply the fact that they emerge on the seafloor and then they try to find a place to land? Or is it
Wait, can you come closer?
yeah, or is it the chemical composition or a geological mineralogy of the rocks themselves? So to answer that question, I have a series of experiments that use 23-million-year-old basalt collected from this site 9 years ago that I was able to get in my laboratory and then sterilize to then bring back out here as kind of a tabula rasa or a blank slate to see how microbes colonize the rocks that are here. And specifically, this would be microbes that come out of the fluids. What we're doing today is setting up the control. So in this rock field, there are no fluids emanating yet. We will get to the fluids for everyone waiting for the octopus action in about an hour or so. And that's where another 4 set of experiments across emanating fluid cracks next to octopus, that's where the other sets of experiments will be deployed. Again, what we're doing here is simply deploying the controls. So the same substrate types. And let me go over my substrate types. The first one, like I just said now, is some sterilized rock from this site collected 9 years ago. Okay? And that's 23-million-year-old rock. The second type of seafloor rock that my colonization exploitments are comprised of is basalts from the other side of this tectonic plate, from the western side of the East Pacific Rise, where an explosion happened 20 years ago. So this variable that my rocks test here is basically the effect of 23 million years worth of seafloor weathering on the changes in the mineralogical composition of the rocks. And we're trying to see if like really old rock, like the one that's here naturally, or really young rock that we've brought from where this tectonic plate is born to here, makes or drives the preferences of which microbes decide to colonize them. So that's the experiment. That's the, I think, purple and green lines that you'll see here that the ROV is deploying now. The other colors are 3 other substrates. The third substrate that I'll describe now is something called mild steel. So we're not sure what's happening around seamounts over the next couple of decades. There's a lot of interest in mining the seafloor because at seamounts, rare earth metals are naturally concentrated because it is water that's literally coming out of the crust, bringing along with it crust constituents such as rare earth metals, which are a really hot topic right now in terms of mining. So I'm deploying sterile shavings of mild steel, which tends to be, along with aluminum, kind of an important component of maritime infrastructure that I'll be able to see, like basically how does this rust, or what rate does this rust, and is that rusting biologically induced? And again, that's my third substrate that I'm deploying here. A fourth substrate is, which one probably the most interesting one for me is something called rhodochrosite. It is manganese carbonate mineral that should allow for manganese dissolution and potential manganese reoxidation after 6 months at the seafloor here. This is our plan, by the way. This colonization experiments are substrates that we are deploying today and will be here until our return to this site sometime at the end of the year. So again, rhodochrosite should allow me to see which microbes that live in the area, right? Primarily the ones that come out of the fluids, are involved in the the metal cycling, specifically manganese oxidation, that appears to be a very important microbial mechanism, or maybe a very, very important microbial mechanism for the generation of these so-called nodules, manganese nodules. So I'll be able to see which microbes carry out this activity by studying their genes on a supercomputer after I come back here and collect this in December, and then analyze their DNA. Okay, one final substrate would be glass wool. I think that's what this is here, if I'm not mistaken. I'm reading GW there, so this glass wool is nothing but an inert substrate. That's like a control for the controls that you
Where are
have.
the markers?
There's no marker.
Oh, no marker.
There's not a marker anymore, yeah.
So we're done. We
Yeah,
can-
so anyway, that's the
Wait, can you
experiment.
introduce yourself before you sign?
Oh, yeah,
before-
okay, yeah. All right, I'll be signing off. Again, my name is Dr. Gustavo Ramirez. I am an assistant professor at the California State University in Los Angeles, and I'm one of the at least 3 or 4 microbiologists, but I guess more of like the hardcore microbial ecologists that's fortunate enough to be here on the Schmidt Ocean Institute's Falcor 2 ship, which is the most amazing floating laboratory that I've seen in my 13-year career doing this. So yeah, thanks to the Schmidt Ocean Institute, and I'm just blown away. Everything here is fantastic. We should at some point tell the audience the experiments that the chef is planning to do with black smokers. It's like even the galley is staffed by artists and residents, apparently. You know. So anyway, signing off.
Thank you, Cas. That's great. So we have deployed these 5 experiments. Thank you. We are picking up our nodule sample, putting it into BioBox 1.
You don't want to put this in a quiver, it's small enough.
No, because it's for microbial, they want it in a BioBox. Thank you.
I'll drop it here.
If we can. Okay. So now we want to wait. If you think most of the sediment has cleared, we want to fire one of the 5-liter Niskins.
Yeah, it looks pretty clear behind us.
Yeah, then that'd be great. Wait, hold on. Let me just check to make sure.
This firebox one,
Firebox 1A. Yeah. And so once that's logged, we're gonna do a Niskin. So tell me when you're ready to log that. Just 1 second.
Yeah, it looks clear behind us and beside us.
Do you know which Niskin you're planning to do?
Probably we'll start with one,
Okay.
the larger one, right? Or is this the
5-liter. The 5-liter. I think
What's your other Niskins?
the other one is a 2-liter later
Yeah, so
on.
5, and then 1, 2, 3, 4 is the 5 liters. and
But I
5
think
is
there's
the smallest.
more. Yeah.
Oh, there's 5 missing one?
Wait, hold on.
Just a second, just a second.
1 second. I just want to log it at the right time, and he's just declaring backlog.
Was optical still back?
Yes.
Three more, I got 3 more. Alright, so this can back?
Yes, please. Okay. We are ready. You can fire that Niskin when you're ready. We're actually gonna do a 5-liter and a 2-liter right here. So. Huh? I'll confirm when he does it. I think he had said 1, but let's just check. So right now we're going to take a water sample by firing the Niskin bottle. So the pilots are just going to hold on to the sort of trigger. Yep, there we go. And that should have closed a bottle of water. So that was a 5-liter, right? Yeah. And so now we want to do a 2-liter. Please.
This will be 1 and 6.
1 and 6. 1 is the 5-liter. Great work, guys. Thank you. Then we're also going to try to do a push core here. And if that does work, we'll do some more. If it doesn't work, we'll just go a little bit downstream, downslope.
I suspect this is going to be pretty rocky, and maybe I'll just top down 30 meters. down
Yeah,
slope.
sure. Gus, is that it? Oh, so where's it going? So they think this is going to be solid, so we're just going to drop 30 meters down slope, or as
I'm happy with
until
this.
yeah, to do the course.
Of course, yeah.
Yeah.
Okay, cool. Yeah. So we're backtracking a little bit, but that's fine. Better backtrack
Yeah.
now than later.
I didn't anticipate it was going to be this
Yeah, it seems
crusty
like, yeah,
already.
It seems like the rocks that we see at the surface are the tip of the iceberg.
Yeah.
We're already on top of a big pile of them. We got a question here.
We'll try to get to like right here.
Manganese accumulation rate, pretty slow.
Where is it now? Oh. Are you? I mean, I know what it is. I know what it is in the CCZ. I don't know what it is here.
I don't know either. Yeah, but say that it's slow.
So I'm not. So there. So with regard to the comment about the rates of accumulation of these crusts, these polymetallic crusts, I can't say what it is here because this is different. But in the Clarion-Clipperton zone to the west of here, where the nodules are not, it's not encrusting. It is solely composed of that. We know that they form at a rate of about 1 millimeter per million years. So if you're looking at a 10 centimeter nodule, I mean, that's Sorry, we're just trying to do the math. Give us a second. Is it what? 100 million years? Yeah, roughly about 100 million years. So these are very, very old. It's basically the seafloor forming again over geological timescales.
Is that right? I don't know.
1 millimeter, 10 centimeter. How many ones? How many? So there's 10 millimeters in 1 centimeter. So 100. Yeah. So 100 million. 100 million. Yeah.
Which is about half the time that it would take the seafloor to get from where it's born, right, the Andes Mountains, to where it's going to die, the Mariana Trench. And that's why the Clarion Clipper,
Clarion Clipperton zone.
That's why it's right there, because it's when they're big enough.
I see.
No, but then
That's how long it's taking the seafloor to generate. So they get to Hawaii.
from the trench,
From
the
Andes
coast,
where they
yeah,
emerge.
right.
And they're halfway to where they're going, which is submersion under Mariana.
I mean, again, knowledge, but there's definitely something about hydrothermal vents involved in the formation as well.
The pads may,
Yeah.
may have
Like-
more heavy metals to be enriched by the zone.
Oh, yeah. I mean, it's where you get like the chimneys, right, or things also coming out of it, coming out of that water stream. And so are the nodules sort of what's left settling out on things, or?
No, it's not. I don't think it's quite that simple, but there's definitely like a linkage in the formation
Yeah.
because they're like not close in proximity usually.
Oh,
Yeah.
I was just curious.
Whoa! Look at that spiky boy. Oh, and an octopus down here. Can we just get a quick zoom on that? This is the first time we've seen an octopus so far off, so far down slope on Dorado Outcrop. I have no idea whether it is the same species as the moose octopus on top of Dorado Outcrop, but we're going to get some great footage right now, quick zooms, so that we can show Janet and Holly when they join us later.
He seems alive, right?
Yeah, definitely alive, but I don't know if it's the same. But first octopus of the day, everybody. And not expected to be down here. No. Well, why? What makes you say that? Oh, no, because that's there. No, definitely not. So this patch?
Yeah.
So we don't go too far?
Yeah, I don't want to go too far. Just
Yeah.
In our experience, especially at the bottom of these slopes, you get
Is it the same?
sliding of the-
I don't know, I don't think she knows actually.
I mean, it's fine. We got good footage. We were just wondering.
Okay, cool. It looked like it was, it was like really not looking super vibrant. That's my scientific comment. Okay.
So next one is fine.
How many are we getting?
And just slowly. Yes.
How many are we getting?
We are doing 8, correct, yes?
Correct, yeah.
And they're all within, once you settle down, all within that little area, as close to each other as possible without impacting each other.
I want to get away from the dust cloud. Just gonna roll because it's the fall one. Come up.
More or less.
I'll turn it.
To the mountain side.
Just where we're looking here. Yeah. Looks pretty good, eh?
The nature of your ocean, you want to know.
once when we were in the Cayman Trench, we deployed some pig carcasses, like literal whole, they were just eviscerated and then whole. And we did not see them again when we went back.
Not even bones?
We were able to find like literally like 1 or 2 rib bones, and it was just by like pure chance. Wow.
Nothing goes to waste.
Yeah, exactly. Do you want to say a little bit about what this is used for? You know?
You want to jump into it?
So we're just setting up to take some push cores of the sediment. We're going to take 8 of them, and we're going to have Gus back to tell us why that is.
Yeah, so we know a little bit about sedimentation rates at this site. So I don't have the numbers specifically, but I know that at least within the first 5 or 6 centimeters of the sediment here, we have it's more or less a record of the last 500 years at least. So there's a couple of experiments that are going to be done with these 8 push cores. I guess I can start describing mine briefly. I'm interested in knowing what microbes live in the sediment here and what they are doing. So to understand that those 2 questions, the standard way of approaching this is doing cross sections of the sediment, which again, because we know the rate, how fast the sediment accumulates, it's kind of a time capsule experiment. In reality, if you go at the bottom of the sediment, you might be you might be looking at microbes that landed on the seafloor as it was maybe a couple 100,000 years ago, you know. So I'm interested in that. So how do the communities survive after decades and centuries and millennia of burial in these abyssal plains? Is a fundamental question that microbiologists know very little about. Okay, so anyway, so I'll be sectioning 2 of these cores and doing the following. From each 1 centimeter section of these cores, I will be using molecular techniques to extract something called DNA. I guess we all know DNA from Jurassic Park, right? Microbes have DNA. I have DNA, right? It's a universal, it contains a universal code that it's the software for what life is, everything in DNA. So that's how I'll be analyzing these communities. It's the new way of doing things in my field, microbiology, for many, many years, right? Since the discovery of microbes and through the golden age, so-called golden age of microbiology, when people like Louis Pasteur were doing experiments, we used to just count microbes and try to assess their diversity, right? The basic ecology, how many species are present in a given environment and what are they doing. We did all that using microscopes, which doesn't tell us a lot because there's 3 shapes that microbes can be, even though within those 3 shapes you can find hundreds of thousands of species. So what we do nowadays is, again, collect DNA and then use supercomputers to analyze the different sequences of DNA of something called marker genes that are analogous to fingerprints. So we fingerprint the microbes by reading their DNA on computers, and then based on the number of different fingerprints, I can generate a pretty good estimate of how many species of microbes are found in each layer of the sediments that we're going to section from this course. Doing a little bit more sophisticated analysis in something that's called metagenomics, I'm also able to sequence all of the genes from all of the microbes that come out of these environments. And this is, again, a little bit more sophisticated, but what I can do there is reconstruct entire genomes of individual microbial species with a very, very high level of certainty. So in addition to answering basic ecology questions, right, like how many different species or different individual, right, unique fingerprints I find in the interface of the seawater and the sediment versus being 16 or 20 centimeters into the sediment, right? In addition to having answers to those questions, after using this molecular fingerprinting technique, I should also be able to generate entire genomes for bacteria and archaea that live at the different layers. And with those genomes, I can then ask who are their closest phylogenetic relatives, who are their cousins basically, and have any of their cousins ever been grown in a laboratory before? And again, that's the real cutting edge in my field, at least, microbiology. It is not just saying who is there and in what relative proportion, right, basic ecology, but going above and beyond that and trying to generate an entire metabolic history and evolutionary history of each one of the organisms that are here by, again, using supercomputers to stitch together their full genomes. So that's kind of the plan or my intention for at least 2 of these 8 cores, and that work begins tonight. I guess I'll introduce my colleague here, Miguel, Dr. Samedo from the University of Porto, to maybe give us a little snippet of what his plans are. He's also a microbiologist. Want me to log in?
Hey everyone, and thank you.
Just come closer.
Hey everyone, thank you for tuning in into this live footage. So yeah, as Gus was saying, we are collecting the push cores, and these push cores will be used by different researchers aboard the vessel to do different things among geologists, microbiologists. In my case, I'm going to be doing some metal exposure experiments. So basically, I'll use one of these sediment cores to incubate with a dissolved metal in order to kind of simulate a condition of...
Wait, sorry, Gus. What's the question about the 1 second?
Is there supposed to be tape on the side of that core?
Yeah, there's supposed to be tape.
Is it clear or is there no tape on it?
Oh no, isn't it there, running down the... You can see them.
Ooh, I forgot to put the tape on my face. Yeah, that's okay. We'll survive. Just go ahead and take it. Yeah. Sure. Yeah. Okay. Cool. Thanks.
Only 2 have holes though.
That's fine.
Yeah.
Yeah. We'll survive.
It's okay. Carry on, Miguel.
No
Good catch,
problem.
pilots. Thank you.
Yeah, thank you. So yeah, so one of these cores will be used for this metal exposure experiment. So I'll incubate a lot of these, subsamples of one sediment core with different concentrations of dissolved cadmium in order to simulate, exposure after sediment disturbance, as in a mining operation, for instance. So yeah, I'll be doing this, and I'll try to quantify the effects of the metal exposure in terms of microbial diversity, taxonomic and functional diversity through metagenomic studies. As Gus was saying, this is kind of a way to investigate microbial communities' composition, structure, and function currently. So yeah, thank you very much. Bye.
Thank you, Miguel. We're getting lots of hearts on the YouTube feed. So we're just going to go through deploying 6 more of these, and then we're actually going to we're gonna do 3 transects as we move up slope again. And that's gonna take us essentially to, much closer to the Octopus Garden. We're gonna transect our way up there.
There was a recommendation from Yerwak yesterday that when the core is put back in the basket, to push it a little bit to make sure
Okay.
the stopper comes in tight.
Ooh, nice big Tina 4.
Oh, yeah, yeah. A little bit more.
You just sit here while I nip to the I'm just gonna go to the back of
Oaken Denise is asking about whether or not the microbes are living under anaerobic sediments, and that is correct, Oaken. Yeah, microbes are quite ubiquitous throughout the entire sediment column. Oxygen should be penetrating here, to some degree, but perhaps not all the way to the bottom of the core. We're playing around with some samples collected yesterday, and the very bottom was 16 centimeters for the size of the push core that we're using here, and it didn't smell like rotten egg at the bottom, so it wasn't sulfitic. But that doesn't mean that there was oxygen, right? After oxygen, microbes tend to breathe nitrate. They can also breathe manganese and iron. And then once those other oxidants have been depleted, they might begin to breathe things like sulfate, which is quite omnipresent in an ocean environment. So long story short, I don't think the bottom of the core here is sulfitic or not sulfitic to the point where I could just sniff it. But it should definitely be anaerobic after perhaps 10 centimeters. And again, that's no problem for microbes, right? Microbes have been on Earth way before there was any oxygen on the Earth. So microbes were running the entire planet when the entire of the planet was anaerobic. So this is a very useful trait. We exploit the fact that microbes can transform sugars, right, in the absence of oxygen, and we use that to make food, right? We use that to make beer. We exploit the fact that microbes are incredible. They have an incredible diversity of anaerobic metabolism. So basically, we use that as biotechnology. So yes, you can be certain that whether or not the sediments have oxygen, there will be microbes that are extremely active all throughout. Okay, so that's looking at the chart. How does it taste? The sediment? Well, I don't know how the sediment tastes. I haven't tasted sediment in a while. Not from this cruise. I did lick a rock earlier, and it tasted like an oyster. Yeah. So one of the rocks that was one of the sounds. So I did smell a rock and did an organoleptic test there, and it tasted like an oyster. I can tell you that. Yeah, other questions here are holes on the side of the push core from Connor. Yes, there's holes. I plan on sticking geochemical samplers there when they're retrieved and collecting pore water for geochemical analysis. Are the holes going to counteract the push core? Yes, they could. Bueno, seguimos trabajando con los sedimentos en el lecho marino. La idea general aquí de recolectar sedimentos es para estudiar las comunidades bacterianas que viven en el lecho marino y en el sublecho marino. Y bueno, pues son básicamente universales. No hay lugar en todos los océanos donde no se encuentren comunidades de bacterias en densidades de los cientos de millones en los lodos que se encuentran en el lecho marino. O sea que es súper importante saber qué están haciendo, cómo sobreviven y qué impacto los metabolismos bacterianos pueden tener en el sistema béntico, en el sistema pelágico y en el sistema planetario. Eso es la idea. Esas son las preguntas grandes que se pueden empezar a solucionar si estudiamos las bacterias que viven en el sublecho marino un poco más. Y bueno, esto es la idea grande de aquí.
Great.
Yeah.
Thank you.
It's a short summary.
Okay, muchas gracias, Gus. So we are eight. And then we're gonna do to get up slope. We're gonna use them as transects, 3 transects. And then we still have spots for 5 non-octopus samples at the top, which we'll do. And I think there will be more corals and other things anyway, so it'll be better.
If possible, you tell them there's xenophytophore there.
And you want 1 and 1?
Yeah,
Sure.
because we're getting to 2 for 1.
Yeah, sure.
Yeah, go ahead, Tony.
Hey mate, just to let you know, I'm just going to move to the west a little bit, just because I want to adjust my heading.
That's fine. I'll give you another waypoint there. Yeah, that's fine. I'll focus right there.
If you could just give the core a little
Yeah, perfect,
to
mate.
push it down, if that's okay. Yeah, perfect. Thank you.
Good.
Oh yeah, there's one right there we can get.
Yeah, it's
Yeah.
it's a good chance to get one of those. Well, the only human debris we found
I
is
know.
from Alvin.
I know. I was just looking at it and I was like, oh.
Yeah.
I actually thought it was ours, but
They don't get lost.
no, we're bad. So in case you're just joining us, while we are currently at a depth of 3,146 meters off Costa Rica, we are at a site called Dorado Outcrop, just to the west of the outcrop, or on the west sort of base of the slope up to the outcrop. And we are currently in the process of taking push cores, some sediment samples, and immediately after this, we are going to be heading upslope towards the Octopus Garden. We're going to be doing 3 transects to get up there. And then, yeah, in about, as soon as we're finished these cores, we're going to be get into the really, really good stuff. This is the Octopus Odyssey Expedition. We are the 3rd shakedown cruise for the Falkor 2, Schmidt Ocean Institute's brand new ship, and it is an amazing ship. We are out here basically characterizing this part of the Costa Rican deep sea where it has been very poorly explored, and many of these sites have never been explored. Doing this characterization is really important for management, potentially, defining human impacts in the future, defining or helping to inform protective measures in the future. And in addition, we're going to be answering some really cool scientific questions about the Octopus Garden, why these octopus particularly like this site for having their eggs. Pardon? Why these octopus like having these sites, being at these sites. And then also some really cool questions about the geology and the microbial ecology of the area. We want to learn as much as we can about the microbes here and what their function is in making this entire ecosystem function. And we want to learn about the sort of the geology and the groundwater, essentially the hydrothermal fluid, where water is emanating from the seafloor and where water is going into the seafloor. Because all of these things together will give us a really good entire overall picture of this part of Costa Rica's deep sea. This cruise is being led by two chief scientists, Dr. Beth Orcutt from the Bigelow Marine Lab in Maine in the USA, as well as Dr. Jorge Cortez from the University of Costa Rica. And this is only our second dive of the expedition. We are out here for 3 weeks. And wow, what a remarkable dive yesterday was. And today has also been an incredibly successful dive, knock wood. And it's going to become even more amazing as we move up slow back into that Octopus Garden. So pilots, for the next 4 cores, for one of them, we want to try to get one of the xenophile cores, which we can see just there in it, please. Oh, you're just too strong. Is that normal?
a break?
Yeah, go ahead, Tony.
Hey mate, yeah, so I'm planning to come round to about 290 heading, so I don't know if you want to drop my position a little bit further south.
Yeah.
So I'm behind you guys.
Yeah, you're gonna go to 290. Yeah, I will put you somewhere. How's that for you right there?
That's all, mate, thank you.
Cheers, thank you.
So we want to get that little xenophile for in one, please. In one of the cores, this little buddy. Yeah, doesn't matter. No, not in two. Given that one's in the ground already. Sure. Yeah. Great. It has it on the top of it, a xenophile for XEN. Yeah, xenophile. Yeah, XENF, I'm sorry, XENPHYOPHORE. Do you want the layout of the whole tray? Oh. So while we're doing these cores, we're just going to do a couple little tests on the RV. Tweaking different parts of the lights. Testing a few things. So yesterday we were able to get some really amazing samples. We were able to collect one male octopus, one female octopus, and a clutch of eggs, which were viable eggs. One of the key questions of this expedition is what species are this octopus? We can now confirm that it was strongly suspected that it was from the genus Musoctopus, but now we can confirm that, though we are still not sure what species it is and whether it is a new species. We also were able to collect a bunch of different corals yesterday. So we collected 2 bamboo corals, 1 primnoid coral. So bamboo corals are isidids, 1 primnoid coral, 2 sponges, including an absolutely beautiful Venus basket flowers sponge. So those are the Euplectella ones, we think. And those are the ones that have these 2 shrimp living within the this amazing glass structure. And what is amazing about this, if you haven't heard this story yet, is that this sponge basically it's basically almost like a cage, right? So it's sealed all the way around, but has, of course, these holes in it, which the oscula, which the sponge uses to function. And so when the shrimp are young, they go into this sponge and use it as a home for protection. A home and a way to be protected. Of course, as that shrimp grows, it gets bigger and bigger, and eventually it actually is too big to leave the sponge again. So you often end up with a mating pair of these shrimp within the sponge, and they spend their entire lives there until they die because they are not able to leave. So we were able to get one of those sponges complete with the 2 shrimp in it. As well as on that sponge, there were a couple anemones actually growing on the sponge. We were able to get another sponge, a bunch of water samples, as well as a few rock samples and some sediment samples. So yesterday we came back with a full tray, and it kept us busy for, a good 3 to 5 hours, depending on which samples you were processing last night. And we're hoping, and so far so good, to again be going back up later today with a basket full of samples that are going to help us to unravel and answer these questions about this ecosystem. And yes, I love what Connor has just put into the chat. I use the word cage, but actually that sponge is actually like a shrimp glass palace, if you will, just one that the shrimp aren't able to leave. But those are remarkable and absolutely beautiful sponges. They are made of silica, and they form the most intricate structures. If you have never seen one, please do go and Google them. Euplectella or Venus flower basket sponges, just absolutely stunning. Yeah, I was just thinking, such a striking difference between the 2 layers. So is one the more like labile stuff at the top? Is that the correct word?
This top's getting chewed up pretty actively.
Right. Whereas the lighter stuff is more compact and sticky.
Let's hope to see more like gradual changes.
Yes, something could happen.
Because you would expect like to see a transition, like a more subtle transition, you know, of the surface to the deep. It's like an event, right?
I think I know who we can ask about this. Really cool. Well, I mean, think about, Jeff about it for a while.
Jeffrey? We.
It's just,
So this is a great time to add that although there's this team of, is it 20 of us, scientists? Around 20 scientists on the boat. We are also being joined online and have been yesterday by several colleagues who are part of the science team for this project, Octopus Odyssey, but we're not able to sail this time. We're hoping that when we have the follow-up cruise in December to continue doing some of this exploration and to retrieve many of the deployments we're putting down, that they will be able to join us. So there's a question about how many samples we can bring up in each dive, and it's quite a lot. So we have space for at least 4 rock samples, 6 different water samples, so Niskin bottles, 8 push cores, and then we've got 3 more water samples which are collected in the vent. Sorry, not 3. 4 more water samples which are collected in the vent fluid. And then we've got 5 biobox spaces that are compartments, which we use to put larger animals or the octopus. And then we've got 8 suction samplers. So when we sort of hoover up any samples, 8 suction sample canisters. So in total, that's 1, 2, 3. 28, we have space for about 45 samples to sit in individual boxes or containers or vessels. But of course, sometimes we do get bonus samples, like you would have seen perhaps already this dive, where we get the nodule that has a coral on it or so on. So this is why, when the ROV comes on deck, the samples on board are able to keep a team of 20 plus scientists busy for a few hours. And we're also able to so for instance, you would have seen just now, Gus deployed some experiments, and so we're able to take things down on the tray and then deploy them, and that frees up space for samples to go back up. Great question, thanks so much.
I'll just bring you to the south of us if you're going to face that direction. Just like that.
Yeah, probably that.
Is the wind picked up or something?
No, it's just the current up here's changing. 1 minute it's down, and the next minute it's from like 310, and now it's kind of coming back. It's 270 again, so that's
Okay, thank you.
How long did it take you guys yesterday?
7 minutes, about an hour and a half.
Okay, that's not bad.
It's not bad. He starts his experiments after we're done processing this.
So right, so you kind of want 1. Okay, yeah, but I thought biology went till like 10:30.
I went to 11 with geochemical putting away everything. 11 or 12.
Holly, in the cool van? I mean in the wet lab? No, wet lab. So all of these samples that we're collecting, both biological and geological, they're going to be housed at the University of Costa Rica, the biology specifically in the Zoology Museum, and the geological in and the geological samples will be housed in the geology school of the University of Costa Rica, and there they're going to remain so that local experts can access them whenever they need to really, allow them to answer pretty basic questions and pretty exciting questions about their country and its oceans. They're also going to be available for experts around the world to request to look at as well. But although we have a lot of work to do when this ROV control van comes up, several hours after each one to process a sample, we usually are just doing the minimal processing that we can. So ensuring that they won't degrade, for instance, like for the biological samples, we'll be putting them into alcohol or to formalin and essentially just preserving them. And most of the work for all of our sample types is going to be done once we get back on shore. And after these cruises, while this expedition is about 3 weeks long, scientists can be working on these samples for decades after cruises sometimes. There's a lot to learn from many of these samples. And in case you are following along on obviously you're following along on the live stream, but in case you're following along on socials, we do have a hashtag Octo Odyssey. Please, if you are active on social, we would love to hear from you there. Also, please do check out the Schmidt Ocean Institute socials as well. The sediment definitely hangs around. Yeah. Exactly. Exactly. Yeah. Exactly. And apparently, like, some of the larger particles settle out like kind of quickly, but like it's the like fine ones just like stick around. So I'm not sure if anybody else in the room knows the answer to this question, but we're having a question from Okan asking whether ROV Falcor is capable of sailing to the Arctic. Can the hull endure Arctic conditions? Is it an icebreaker? No, just ice strengthened. Yeah? So I think the answer to the question, though we will definitely clarify, is that yes, it is capable of sailing to the Arctic, although it is not, certainly not an icebreaker. And we're double checking whether it has an ice strengthened hull. Gus, do you know, Gus or Miguel, whether previous samples, whether we were able to sample vent fluids before at this site? I know definitely no biology, but were geology or water samples taken before?
I think it has been tried, but I'm not sure to what level of success. So
Okay.
When Tim is here, we can talk to Tim about it. Tim is on the orchid and wheat team. I don't know, yeah. Regarding the sampling of vent fluids, it's been attempted before, but I like to remind the audience that humans have now visited this site for 9 years, right? So 9 or 10 years.
Well, 2013 and 2014.
Right. So the last time that scientists visited the site was 9 years ago. So I'm quite sure that a team of geochemists, more than likely led by Dr. Jeff Wheat, University of Alaska Fairbanks, located in Monterey, California, he has attempted to collect pore fluids. I'm not sure if that was successful or not. It's quite complex to get a pure sample of pore fluids, just because those fluids are simply emanating from the rocks where the octopus hang out, and then immediately, of course, blending, right, or dissipating with the rest of the ocean around them. So I'm not sure if they've successfully have done that yet. What I do know is that is being attempted on this expedition now. So Dr. Beth Orcutt from Bigelow Laboratories and Tim D'Angelo, Dr. Tim D'Angelo, who is also from Bigelow Laboratories, have something called geochemical squeezers, correct? They're very sophisticated syringes built by the engineers at the Monterey Bay Aquarium Research Institute for them, that basically allow them to have a giant syringe that's amendable to manipulation by the ROV arm. So I think there was 2 attempts yesterday to fill the syringe right at a site next to a mother octopus where there was shimmering happening. So I will put Dr. Tim D'Angelo on the microphone as soon as he comes in here, but I know that we've potentially done that already, but that's just in terms of collecting a sample, not necessarily doing the geochemical analysis. I guess another thing that has been going on is that it seems that there was something called an osmotic, an osmo deployment here. So what is this? This is another technique used by Dr. Jeff Wheat, again from the University of Alaska Fairbanks, that employs a semi-permeable membrane across a pump that's made out of half brine on one extreme, then the membrane, and then DI water. And all of that is connected to an extremely long 1 millimeter thick coil of tubing. This tubing is connected to the pump, so everything's filled with water when this is brought down to the seafloor. And the surface area of the osmotic membrane is tuned so that at the temperature of the seafloor, we know exactly how much water comes into a very, very long, thin coil of what essentially looks like aquarium tubing. We know how much water is brought in by osmosis as a function of time. So I believe that these things are calibrated to bring in water that displaces the DI water in this thin tubing at a rate of half a meter or a meter per day. So if many, many meters, perhaps hundreds of meters of aquarium tubing are deployed here, you can have a time record of the geochemistry, especially if the inlet is placed exactly where the venting fluids are at. You can have a time record of the dynamics of the geochemistry after these samples are collected, many days, perhaps years later after the samples are collected, and then sectioned 1 meter at a time. So that's an ongoing experiment, I believe, that's been happening here for a very long time, perhaps 10 years if I'm not mistaken. But anyway, hopefully that clarifies the question regarding the collection of fluids. Or kind of a long-winded answer again for me, but I hope that that brought the efforts that are ongoing and have been tried in the past, the efforts towards
All good?
exactly that question there. That's what we've done to address the collection of fluids here.
Oh, yeah, we still have to put a couple of counts on.
So we are nearly finished stowing all of our push cores. You got to secure them because the RV has, a long way to go. We're down here for at least another sort of 8 hours. So we need to make sure everything's secured. We certainly don't want to leave anything on the sea floor. So we're just wrapping that up, and then we're going to begin our transit up to the Octopus hotspot area. On the way there, to maximize time, we're actually going to do 3 transects up there to get us up there so that we're able to use that footage for community analyses in the future, but also it's part of our journey to get up to the top. Alex, there's a question in Facebook I don't know the answer to. The last one.
Yeah, just on Facebook. Previous samples.
Yeah. I mean, definitely give a hail out to previous dives, but.
Yeah, I'm trying to think like how so. Yeah. It's too short. Let me get an answer for that.
Sure. Is it time? Nearly. Okay, so let me tell you where we are. So we are just stowing the push cores. We just need to put a few of the little liddy things on and then pick up those 2 pieces of trash as well as one of the lids and bust it off. So once we're done all that, then we are good to, we're going to transit from where we are now up to the Octopus area. But on the way up to waypoint 3, we are, which is in the rocks, we're going to do 3 transects, 100-meter transects. And so just like kind of, I guess, just like zigzag your way up there. Yeah, 3.
Okay.
The settings are all here, but I can wait until, if that's helpful. And I think on those, we're not doing any sampling, any zooms, anything. We're just doing the transects.
Yeah, yeah.
And then when we get to the top, there's this list of animals that we did not collect yet, but that we might want to include somewhere along here when you have time. So we are just about to do a science watch change while the pilots are continuing to stow these push cores, and then we're going to be heading up to the Octopus Garden and into some areas with some amazing other biology other than the octopus, which of course everyone's actually here for. Anyway, thanks guys. Actually, maybe I'll go eat. Did you eat? Yeah. Okay.
Yeah.
Okay, then I will.
Yeah.
No, I'll wait till later. No, because I want the lunch finishes at 12.
No, you're.
Oh, yeah, right, yeah.
So you have to stay.
I'm ready. We're nearly done here anyway. We're going to do 3 transects up to the Octopus Garden. So use the journey up there to do transects.
Everybody's waiting for it.
Exactly. Everyone's like we're like, cool, sediment's cool, but
Yeah.
we're here for the animals.
Actually, I'm happy that today I got to see the segment because yesterday
You were asleep.
yeah, so this is nice.
I'm supposed to text everybody back home when we're at the Octopus Garden.
On this data sheet, I had a lot of like additional things I can add on some things that I took. What should I do it on here in the notes or?
So there's 2 sheets.
Oh, no,
So there's so that's the master log. They're not putting anything extra onto that one. But then if you go into protocols and macros, then that's the macro picture database.
So we have a question over on Facebook asking, is there any place online to see previous samples taken? And as far as seeing the samples themselves in person, those would be at the institution that houses them now, whichever that might be. But if you go to the cruise page for any given SOI cruise, at the bottom of that page, there's a data and publications section. And that has some samples, or if there's been any published data as well, you can find that there. So with this cruise still happening, you might need to wait a little bit.
Oh, we're doing an ROV dive change too. Duh. Okay, well then I'm definitely.
It's in there. Yeah. How many of them is there? I just did the covers, is it?
Lunch ends at 12:30.
Where do you put that?
Yeah. I know, but you can go.
Once they come in, what?
Something different. Because it's
Right, so let's write it onto here so they also have it. Sorry.
Oh, like 3 transects. No problem.
Just waiting for this handover right now. Scientists sharing notes, and pilots are also doing the same.
It's actually a little bit downhill from waypoint 2 to waypoint 3. And it's 100 meters, and they're gonna like
So they're all waypoint 2, waypoint 3. The only way,
they're
the only good way
basically
to tell
going
when we have the
up slope. So they'll continue, so one will continue from each other. Basically. Does that make sense?
There is other people on the board in the lake, but is there the lake jam also?
No stopping, no zooming. We're literally just driving.
Let's just monitor that.
It's connecting transits. And then when we get to the top, all the connections can happen.
Let's think about the distance, you don't have to write it down anymore.
Where's the camera?
Or
We
fire.
asked the pilot. So when normally we set up with pilot support.
If I tweak them, I can get them all to do that.
And
Yeah, 'cause that one, American highlights this.
There's one yesterday, Christian just took a good on our cushion, but then it went fine. But then it's not always the same. I think it's how they're set. Yeah, sometimes.
Well, then, there's different angles on them, because they come around, so you want friction and
Yes,
lines.
yeah.
So right now,
I'm
we
just gonna wait to
are,
set up the transits.
down here, we're gonna go back up to this nodule field.
Oh, I see.
And do some sampling, and then.
no, we go
Yeah.
we go to the rocks
Oh, marker 3.
and we do the transit. So it would be good, Mike. You're gonna be the pilot. You're gonna be the pilot.
That's like.
For anyone just joining us online, we are coming to you live from research vessel Falcor 2, owned and operated by Schmidt Ocean Institute. We're currently looking at live footage from ROV SuBastian, about 3,100 meters below the surface of the sea at Dorado Outcrop. We're in the middle of a handover right now, and then we're going to be heading back up to, we call it a nodule field, but we've since discovered it's actually a little bit of lava, I believe. And we are going to then head up this outcrop where we saw many octopus yesterday.
Yes. And it's just like the last ones, the transit's gonna follow one after the other. So it's basically a random heading up the slope, another random heading up the slope, another random heading up the slope. Just follow this continuous 100-meter connected. But it's okay at the end of them to have like end your transit, do whatever you need to do. But then from roughly that spot, continue in another heading. Alright, yeah, it's busy. You got to go up to
That would be one of the tricky things to disappear during that transit. So we move to marker 3 in the rocks. There we'll do that with 100 meters and an angle of 100 meters and another 100 meters and continue.
Okay, yeah, so. Not following the line, so you're wanting to.
Yeah, like the first 100 could be following the line, then like a little angle,
Come off and then
then
slowly.
yeah, and then following the line again and continue.
Okay, yeah, so just sort of zigzag, 100-meter sections just to cover
Yeah,
all the areas around the line.
it's only three we need. So and it's important that once we start, say you set it up and the speed is right. You say, okay, start. So we start and just go with the lasers on. And when we have 100 meters, give us a sign. We stop the recording there. And then when you're ready for the next one, we do the same. So the idea having the lasers so we can see it's about 2 meters that we're looking at. So as we go, we can start counting. So it's not only there's this bunch, but there are 25 per square meter. We can quantify. And then once we get to the top there, it's the whole, we do some collecting.
Something everyone needs to work on. Only confidence should be not.
Yeah, I see that. Yeah, I saw him
Yeah,
yesterday.
yesterday was very exciting.
That's good.
There was a
How long have you
question
been in this? How you been up?
12 years I've been doing ROVs, but this is only my second science.
Oh.
I was oil and gas.
Oil and gas previously.
So, and I was like,
Well, many have experience from oil and gas before.
No, I'm still trying to count.
This must be very different.
Yeah, very, much more interesting. I much prefer it.
Oh no,
That's good.
The person was asking specifically about having a sample in hand. Was there
Yeah, once in a while I go with a tourist boat to an island of Costa Rica, it's Octopus, and to do science. And they always have this. We love this. It's like out of the routine and seeing interesting things.
So we're gonna put it in that bio box on the left. It won't take up much space, and it shouldn't affect any samples we take, but you guys okay with that?
Yeah. For deep.
More than I was expecting.
Yeah, let me check.
Yeah, I'm actually very sad about the lack of widespread
I mean, you just haven't been here the right meals.
Well, I tell you what.
They only have like
I think put them those,
soft areas.
because those will be for the octopus like this.
Chip.
But we haven't been back.
Okay, we can do that. Let's put them in the back. That's it.
With the bio boxes in the back crate.
They'll fly out of the crate, I think.
So, porch out of it. You move your arm round a bit, just by porch out, so make sure that catch in is kind of close to the bottom of the arm there. That's us through out.
Wait, does that mean I'm the
What's that? Two?
Okay, then I'll follow that.
Yeah.
Actually, no. So, we'll schedule the science part meeting. Once the ROV is back on.
If we put it in the other ones, I'm worried
So
about
like
that.
8:30.
Deploy with the two pods. Alright, we'll get it back in there. I'll let him toss it in there, and if I have to, I'll push that movie out there.
Well, then it's caught enough of the lid there.
Yeah, right, go for it.
Should open it up.
Isn't that why you get to kind of, because like it's just one link always for their streams. Here it's like, I think I noticed yesterday there were 2 links on YouTube. Sequential.
And that's because I have to check like a top office and know how back certain hours in their stream. If you keep a YouTube stream under 12 hours, you can go back all the way to the beginning of it. And as soon as you're not live,
So it's pointing away, it'll point towards the box.
but if you go over 12 hours, then you have to pick up these things, you just dispose of it. And so you have something they keep their own recordings.
Yes.
No,
Yeah. I see. That's helpful, because I was like, this is so unhelpful,
Yeah,
because
so
every
you're in a
morning it's
position
my
like
mom's
this too.
like, mom, super savvy, and it's like send her a new link, to be like, this is the link. So
Now, that being said, there is a static reflection always used.
You're already pretty much there, so
It's like,
if you're above it, I would just angle it a little bit more. And, got it.
check for a second.
Okay, one more. G.
It's being placed here.
I'll close the bio box and stuff.
What does this get categorized as a problem?
oh, so that'll take us to our chain.
Right. Like the one that says lunch, circle here.
Yeah, there is a line.
It gets categorized as a problem.
Well, it's just kind of an alert saying this is in us.
Okay.
So when we open it,
Yeah.
it'll be like, what's this?
Yeah. Do you mind if I just move your brain on this?
Just
Okay,
sit
so
on
some sample numbers.
anything unexpected.
All right, for anyone who jumped in just in the last couple of minutes, saw a question of what are we doing right now? We are towards the end of a handover. I think the handover's mostly done, and we're picking up some bits from this push core sample area that we were at. After this, we'll be making our way back towards the outcrop. We're going to swing back by the field of, well, I guess lava now. And then we're going to head up the outcrop and look for more octopus.
So if it's something special, make sure it gets here this way.
If I see heap, no.
Yeah.
Fair enough.
So like, this was octopus.
So we're working two,
Like, I know that already matches up, like, I just say it's octopus.
is there any
I
geophysical
really want to meet
character
these.
you can given the slope
Is the egg
with their new turbidity
Right, so the
curves
whole egg
coming
white.
down.
But then if you were doing each
I mean, you have to have some sediment sitting on top, so.
I don't do that. So
It's
what I
such a
do
contrast
instead
between
is
what
have
you
a
see
description
on top
of
and
the
then down.
No, I'm
samples
afraid
that
it's
I
frost.
took, and they don't have
But then
those
that's,
unique
our focus
numbers
locks down, it's just the rocks.
I understand why like that's the important thing. But if you think about like what that would be important for, it's like in the publications or in a museum discussion or wherever it is biological go, all you actually really need to know is that it came from this much. And if you do start working with an individual, yes, then that's what I did. I would change this. Like these should maybe just all be one.
Yes, because they're subs.
Right, but there is not a sub-sample.
No, so then, yeah, exactly. These
Like
aren't
this is
sub-samples.
this
These
is
are-
redundant.
So you don't actually, if this is just for your clutch,
Yeah.
then you don't, so for instance, so like our riser one, we don't have any of these of the force sample, because it's just a whole sample, there is no sub-sample.
Yeah.
So I think that's how your stuff would be,
Yeah.
unless you decided to do each of your.
And and I don't really want to do that. So I can leave this all as full
No,
materials.
so I would, so
Not have a sub-sample.
unless you want.
But the thing is, I want that for myself. So what I want is to have
The
this.
sleep
I want to have
lemurs,
it.
three got kids.
But it's just not considered a sub-sample.
So what I would do then
You
is
know about them.
difference, and you have.
I wish that we could just change or add a different. I guess this sub-sample labeling scheme actually does.
Yeah, and so that's why I think it's okay just.
Could we do like sub-sample for like what individual?
I would, no, yeah, no, so I would, the only thing I would suggest, you know, if you want to include things on this sheet, is create a new column, and you have like. Octopus. Subsamples, and you do your own scheme for your subsamples. Rather than, because I think if we put it into this one, it will confuse people.
If you were with us early enough in the dive yesterday, last dive, you know that we had a little bit of a fight with the sediment here. It's very slow to disperse. And so we're just kind of waiting out a little sediment right now, and then we'll be underway. After we recover some of our items from the floor here.
next thing he's gonna watch out in that box. We gotta get that flag, marker. Yeah, just see what you can do and we'll close it. Just communicate with Tyler there. That's not actually on the information over that. So you got heading to station cruise.
Yeah.
Yeah. So you can do like right here too, positioning.
Okay, and that was noted down, right?
Right there.
Okay, well then, if it's gonna be long, I only have 10 minutes. So I was trying to wait for the start of the transects, but I'll be back.
Enjoy.
Thanks, I'll be back. I didn't think you would.
I'm not that.
Yeah, that sets that lot, that locks in your, this is what you're on, position. You're not going anywhere now.
Yeah, I'm gonna get it.
Alright, you ready for it?
No, probably.
We need to work that out.
They're counting more than time, so I don't know.
Gotcha.
I'm hoping that I can have it done before my shift starts at 4.
Okay,
Yeah.
Sorry guys, just find this, view viz a little bit.
That's slow.
I think I would actually just, I would just grab the tip of that. Yeah. And just try to hold it under, and then close it a little bit better.
You can see that one of the challenges of working underwater is when certain things float. So this is a little flag so that we can mark a location and return to that in the future, but mark it actually with a physical marker. And the flag just wants to float. So we're trying to tuck that back into the bio box here for transport.
Back out of there, back around, close. It's in there, it's not coming out, so we're good, let's roll. Okay, so yeah, just stay clear. He's gonna pull the portsman, and then what you wanna do is stow it. I'm sure you kinda know the position. I'll put you a little bit close. In the meantime, you can stow this right to your right, because you want this arm straight out. So about 45 degrees in, so just start shouldering right.
All right, and with that, we can tuck the porch out of the way.
You go more resolution, pull back, so go lower. Exactly. Freeze it, pull it all the way up. Go, all the way. Right. Yeah, just like you're doing, all the way in. Okay, now that joint is pitching, so it's not poking out there. And perfect. Freeze. And exit. Exit. Now you're good to start. So set it like this. Yep. That's not. Thanks for the yeah. Okay. Back to the
Let's go to no, we'll go to that green square,
can't really think of this.
and we're interested in the rock, solid rock.
Yes, this.
So yeah, so go to that green square. We'll see how it looks. If it's okay, then we'll get ready for the transit.
Slow as fast.
Slow as good. Slow as fast. Slow as fast. We're on island time, right? It's on the
When do we begin the transit?
We have to get to the green box.
The babies? Oh, no, the waypoint.
Yeah.
Okay.
We're down here. To get there, we'll see how it looks if it went into the rocks. Then we do the transits, then move up.
Okay.
There's those still sitting on the transects are from those green boxes.
We'll see what it is under the green box. If we're on the rocks, then we'll start the transits. I'm not sure if we're going to be there or just short of them.
I think we'll see rocks then.
Yeah.
All right, I'm going to put the map up for a moment here, just so you can kind of see where we're heading. A little small in the corner there, but we are going to be heading up the hill now, up the outcrop. Now this is kind of where, right as we started up the outcrop yesterday, that's where we were encountering the octopus.
It was off while it was on. And killed the captains. Title tail. That's bad. Let's just monitor it for now. If it gets worse. Fix that. You zoomed in on the 4K? Zoomed in on low. No, just to get this stuff. See if you get that. This cloud, we're clear to start moving.
All right.
Okay, we're on the move again, and we're going to start the afternoon session. My name is Jorge Cortez from the University of Costa Rica, and with me is Rob and Janet helping during this next 4 hours of this expedition. And we're going to move into the rocky areas and start doing some transects. I'll explain later on what they are, and looking at organisms and trying to collect some of them, including some of the octopus. And sorry about that dust storm we had. Muy buenas tardes, ya estamos listos para continuar en la sesión de la tarde. Yo soy Jorge Cortés de la Universidad de Costa Rica y ahora nos estamos desplazando hacia el montículo rocoso. Y ahí vamos a hacer unos transectos, ahí voy a explicar qué son los transectos y posteriormente vamos a recolectar organismos en esa sección. Aquí esto es una región que ya habíamos visitado en la mañana y lo que vemos ahí son corales negros, eso parecen abanicos, y sobre el fondo vemos también estrellas quebradizas o fiuras, y esas rocas son unos lóbulos de aparentemente basalto. Esto está en estudio ahora. It's better.
And on the SOI side of things, my name's Alex. I'm the multimedia technician on right now. In the pilot seat, we got Mike, and co-pilot, we got Tyler. And Chill is also here from the ROV team.
What we'll be doing is
getting there, we'll see if we're in the rocky outcrop. If we are, then we're going to do transits. So we'll be 100 meters at a point 1, is it? Yeah, 0.1 meters per second.
Yeah.
At a meter from the bottom.
Right.
Trying to be safe.
Yeah.
And so we'll mark the beginning and end, and then we shift a little bit at an angle, do another.
That'll be at marker 3, right? We'll shift.
Well, I think we, well, once we get here, because that's less than 100. I mean, it's much more than 100.
Let's see here. Let's measure. So you'll want to stop somewhere around here.
Yeah. Then what we would do is maybe another one like this at an angle.
Okay.
And then back there.
Is that the only transect you want to do?
There are 3. Yeah,
Three.
that's the indication. 3 here.
A random angle after we hit?
Facing down the hill. You want to be another angle that is on the same down an angle on top of it. So we do that if we
Go over the top.
zigzag up the hill or maybe come back down and do another.
Yeah. So it'll depend on how steep it is.
That would be another option. Once we get, we'll have to see if it's rocky out here, we can start there, or we can do it like this.
That could be better for us. What he's saying is, if the hill's this way, we can fly up the hill, no problem. But if you have us turn backwards, the RV will be much higher and you'll have no visual.
So let's do it that way. That's right. Okay.
That's exactly.
Do it like this. And it's free then.
All right. Okay.
And
If folks tuned in for the dive, our last dive, you might remember a large yellow sponge. Just looking at the map right now, it looks like we might pass over that again. So if you look, we've got it marked up there as a marker. So keep an eye peeled for that.
Yeah, so just right at the edge where we've got it just now is just at the edge.
Yeah. No zoom.
No zoom. Well, we've got a tiny bit of zoom just to get.
Right now, Jorge and the pilots are kind of going over the plan for what we're going to do. Going to head up the slope, and then, they're talking about where we're going to, what we're doing when we get there. Lots of planning.
That's what they want right there. You want that and no zoom?
Yeah, no zoom, and we will need the lasers. See, there's a shadow now. There's like a shadow here. Can there be removed? This morning I think it was more to the side.
Pilots are currently going through the lights. If you were with us a few minutes ago, you might have seen some flickering going on, so they're just checking out what's what.
So now we're getting close to the rocks. Will we just go up a little bit more?
it's out a little bit, but
I see there's a question about the octopus collection from yesterday. We do have Janet here. I'll try and get an answer in a little while.
Where's the question?
The question of how the octopus looked when they got back up. Were you impressed with the samples there?
They were just beautiful. That's all I can say. They were like so cool. Like 2 hands full, muscular, broad, I mean, meaty octopuses.
Nice.
So people think that if you bring a deep sea animal up from the bottom to the top, it explodes. But that does not happen unless they have air inside of them. Many fishes have an air sac that gives them buoyancy as they move up and down. Rat tails, I always forget the real name of rat tails, they have that. But octopuses are solid, fluid-filled muscle. And water, which is fluid, is incompressible. So they're the same on the surface as they are at depth. Air is compressible, which if you go in a cheap airline like I fly, you got a little package of crackers, that package balloons up at the low cabin pressure. Then when you land, it's normal again. Air is so fickle.
Yeah. So fortunately, the octopus didn't have to deal with the fickle air.
Here we're seeing the green circle there, that's a sea urchin. And further up to the left, center up is a sea cucumber. Estamos viendo un erizo de mar y un poco más arriba un pepino de mar. Y seguimos subiendo en la formación rocosa que hemos estado estudiando. Esta es una de las especies típicas de peces que vemos en estas profundidades. It's one of the typical fish we see at the depths. We're at 3,000 meters. Mm-hmm. Mm-hmm. Great. That's good. Looks good. And with the lasers, then we can figure out how wide it is, and then how long we go, and then we can calculate how many organisms per square meter.
So if we're flying, do you want to fly a zigzag pattern? Do you want us looking at the face and zigzagging like that, not following a line?
So they're currently working out the details for flying some transects here. As you heard, of course, we have the green lasers on. So right in the middle of the screen there, you might see 2 dots. Those are about 10 centimeters apart, or they are 10 centimeters apart. And so if we keep those on while we fly these transects and we look for the organisms that we can see, then we can sort of figure out a density.
Yeah, we'll go up a little bit more. There's still too much. Lots of sediments.
Just waiting for the vessel to catch up just a little bit.
Mm-hmm. It's surprising the amount of sediments we see here because we are way off the coast. So the terrestrial influences must be low, but we see all these sediments probably from the productivity in this area and just rains down and in here. See another one of the very interesting sea cucumbers. How about there? Better? Yeah, okay.
Jorge is soft-spoken, but he carries big stick.
Whoa, whoa.
Every word matters.
Okay.
So do you like this to start the transect?
I was thinking maybe when we get it gets denser. A
You
little
like
bit
more
denser.
exposed rock?
Yeah.
Okay.
So let's try to get to that square if it still looks like that.
Come some of the way.
Mhm.
So turn those off and
Yeah, I've seen here some very interesting sponges.
Imagine you were a gravid female octopus struggling up this hill looking for a clean rock.
Yes.
It would seem like forever.
Right.
Maybe you try a little crevice here, a little crevice there, and not find the spot.
Well, it was interesting. It was interesting yesterday where next to the octopus that we're brooding, you could see like old glue where clearly this crevice has been used by octopus after octopus.
In 2018, we published a paper and we noted that the same spots are used over and over again. Nobody knows how long that glue stays, but we were seeing no development in any of the eggs, and we were thinking some in that fluid is doing them in. So, and then yesterday I was just stunned by what I saw. Totally different. And a lot more octopuses, it seemed to me. But once you're stunned, you know, things pile up. So I'm glad we're doing it transect so we can do this more quantitatively as opposed to, oh, look at that, let's go over there.
Yeah.
Which is the fun one.
Let's move up a little bit more.
We think when the octopuses themselves aren't keeping the sediment free from around where they're sitting, but it's the flow of the fluid, the shimmering water coming up from the crust that blows that sediment away. That's why there's a dark spot around the octopus. I think of it as a little halo.
tilted this one up just now, so this is what's coming up before
Okay.
it reaches the
Okay.
side of this
See,
tower.
it's getting better.
There's a
Oh.
pod.
There we go.
No pod.
Uh-huh.
Picture.
Yeah, we see one of the octopus,
Female.
a female octopus.
All right. Well, it is getting close to the time I need to reset streams. So I'm going to do that before we get like hip deep into octopus. So I'll be right back. Just sit tight. New streams are going to pop up on YouTube and Facebook.